Good evening, students. I hope you have done very well for your NEET exam. Now we'll go ahead and do the discussion of your botany paper. Okay, so let's go to the first question. Okay, the first question is identify the set of current statements. The first statement is the flowers are of Vallisneria are colorful and produce nectar. Vallisneria is a aquatic plant. It will undergo hydrophily. So that statement is wrong. Flowers of water lily are not pollinated by water. That is correct. Usually with insect only the pollination happens. And the most of the water pollinated species, the pollen grains are protected from wetting. That is true. And the pollen grains of some hydrophytes are long and ribbon-like. That is also true. In case of Zostra and all, it will be ribbon-like. And in some hydrophytes, the pollen grains are carried passively inside water. That is correct. So the correct option would be B, C, D, and E. So the first statement is wrong. Vallisneria is an aquatic plant. So that is a wrong statement. Other than that, all the other statements are correct. Okay. So the option for the first question will be 2. Let's go to the next question. Okay, the type of conservation in which the threatened species are taken out from their natural habitat and placed in a special setting where they can be protected and given special care, that is called as biodiversity conservation. Okay, so this comes in the ecology chapter. This is a fact. The next question, inhibition of succinic dehydrogenase enzyme by malonate. It is a classical example of this will be competitive inhibition. So succinate dehydrogenase enzyme, if you see, if it has a structure like this, we have two components which are present here. One is succinate. We all know succinate is a substrate. And then we have malonate, which has a similar structure. So both will fight for this position. And this, if malonate goes and binds, obviously the substrate, that is succinate, will not be able to bind. So the answer for this one will be competitive inhibition. So the next question, identify the part of seed from the given figure which is destined to form root when the seed germinates. For this, if you see here, this is the structure of embryo. The above part is this coleoptile and the below part is the coleorhiza. This is the radical part. So the option for this one is C. That is, radical will be the correct option for this question. Bulliform cells are responsible for. Bulliform cells are special cells which are usually present in the monocots. They help during the water availability when they are less. So it will be helping in inward curling of leaves in the monocots. Which of the following are required for dark reaction of photosynthesis? The Calvin cycle. So here, what are the things which are required for Calvin cycle? We obviously require carbon dioxide, NADPH, and ATP. The chlorophyll pigment and light, they are responsible for the light reaction. So here the option will be C, D, and E. So the next question, formation of interfascicular cambium from a fully developed parenchyma cell. That is an example of, we have studied about this. That is, if you have a meristem will differentiate to become permanent tissue. And it will de-differentiate to become secondary meristem. And it will re-differentiate to become secondary permanent tissue. So the answer for this one is interfascicular cambium. So interfascicular cambium is in between the vascular bundles. that is the medullary ray, that is the one which will undergo the change. So this will be dedifferentiation. Dedifferentiation is the answer for this question. So the next question, that is Hind2 always cuts DNA molecules at a particular point called recognition sequence, and it consists of, for Hind2 it will be 6 base pairs. Usually it will form a blunt end in this scenario. So the next question, tropical regions show greatest level of species richness because tropical latitudes remained relatively undisturbed for millions of years. Hence, more time was available for species diversification. That is correct. And tropical environments are more seasonal. That is wrong. So, when we take the surface of Earth, so near the equator, we say it as tropics. And above, we say it as temperate. And below we will be saying the polar region. So usually in the tropics, it will be summer. Summer and heavy rainfall will be there throughout the year. But in case of temperate only, we will have 6 months of summer and 6 months of winter. Summer and winter will be there in temperate region, whereas in tropics, it is mostly stable. Throughout the year, it will be stable. And more solar energy is available in the tropics. That is correct. So more to the equator, it is more sunlight is available. Constant environment promote niche specialization. Yes, mostly it is summer, so niche is also specialized in this region. And tropical environments are constant and predictable. There is no much change in this scenario. So the correct answer for this one will be the first option, undisturbed, and then B are more seasonal. That is wrong. More solar energy is available. That is correct. Constant environment promote niche specialization. Promote niche specialization. That is correct. And tropical environment are constant and predictable. So the correct answer for this one will be 3. So இது வரைக்கும் எல்லாருக்கும் நல்லா புரிஞ்சிருக்கும்னு நினைக்கிறேன். Okay, we'll go to the next question. So, which of the following is not a criterion for classification of fungi? So, in fungi, we classify them basically based on the reproduction. What is the type of reproduction? What is the type of spore? That is ascospores or basidiospores, and the type of fruiting body. So, here the nutrition is not the mode of classification for the fungi. So, next question. So, how many molecules of ATP and NADPH are required for every molecule of carbon dioxide fixed in Calvin cycle? So, for every molecule, that is for one carbon dioxide molecule, for one cycle, it will be using 3 ATP and 2 NADPH molecules. Okay, for 6 cycles, that's when it will become 18 ATP and 12 NADPH molecules. This is based on the photosynthesis. So the next question, these are regarded as major causes for biodiversity loss. They are asking for the correct option. We have studied about the evil quartet. So evil quartet, obviously overexploitation, co-extinction, mutation will not be a scenario, habitat loss, migration will not be a scenario. So the correct option for this one will be A, B and D. Okay, so this comes under biodiversity chapter. So we'll go to the next question. The capacity to generate a whole plant from any cell of the plant. This is called as totipotency. So usually in plants, we have studied about tissue culture technique. So in tissue culture, this is what is used. That's when we are able to take any part of the plant. That is, we can take a leaf or a stem or a root and we can propagate into a new plant. So that ability of the plant is called as totipotency. So the next question, equation of one Pearl's logistic growth dN by dt is equal to Rn K minus N by K. From this equation, K indicates the carrying capacity. Carrying capacity is the correct answer for this question. So, spindle fibers attached to kinetochore of chromosome during. So, we have studied about the phases. So, in prophase, what will happen? In prophase, the chromosome formation and the nuclear membrane degeneration will be happening. And the next phase, that is the metaphase. This is the phase where the metaphasic plate arrangement, that is equatorial plate, it is called as. So here only what will happen? The chromosomes will come to the center, and this is when the spindle will attach. So this attachment and coming to the center happens in the metaphase. Okay, இது வரைக்கும் எல்லாருக்கும் நல்லா புரிஞ்சிருக்கும்னு நினைக்கிறேன். Okay, so இது வந்து செல் சைக்கிள் சாப்டர்ல இருந்து நம்ம பேசுறோம். So metaphase, இதுக்கப்புறம் தான் நமக்கு வந்து we'll have anaphase and telophase. Anaphase தான் the separation of chromosomes will be happening, and in telophase they will again the karyokinesis will initiate. So the nucleus will separate. So we'll go to the next question. Okay, so identify the type of flower based on the position of calyx, corolla and androecium with respect to the ovary from the given figure. So if you see here, this is the inferior ovary. Okay, so this is half inferior. So this will be second perigynous scenario. Okay, perigynous, we have studied about this in the morphology chapter about the half inferior, superior and inferior ovary. Okay, so for this, it's about fungi from biological classification. So Rhizopus is commonly called as bread mold, and Astilago, this is commonly called as smut. Astilago is called as smut. Puccinia, this is usually called as rust, and Agaricus will be mushroom. So based on this, the correct answer for this one would be 3. The correct answer for this one would be 3. Okay, so next question. Okay, in a plant, the black seed color is dominant over white seed color. In order to find out the genotype of the black seed, with which of the following genotype will you cross it? So what they are saying is the black seed is capital B, or it can be capital B and small b. So how would you know whether it is this one or this heterozygous scenario? We have to do the cross with recessive, that is with small b, small b. This is called as test cross. This is called as test cross. When we do this cross only, what will happen? If you are getting capital B, small b, only this, then we can say it is a dominant. When you do a cross with heterozygous, we'll get capital B, small b, small b, small b. That is 1:1 ratio. So with 1:1 ratio only, we'll be able to confirm that this is a heterozygous parent. If 1:1 ratio didn't come, then we'll be saying it as homozygous parent. So this cross should be done by the recessive parent. Small b will be used. So let's go to the next question. So a pink flower snapdragon was crossed with a red flower snapdragon. What of the phenotype is expected? So a pink flower means you will have this, and red flower will be this. So in this, when we make a cross, we'll get capital R and capital R, small r, because obviously this is homozygous, so we'll consider only one of the alleles. So red flower as well as pink flower, we'll get the ratio 50:50. So the answer for this question is 4. So this question, 2 or more alternative forms of a gene, that will be allele, cross of F1 progeny with homozygous recessive parent, that will be test cross, cross of F1 progeny with any of the parent, that will be back cross, number of chromosome sets in a plant, that will be ploidy. That is, we can say haploid, diploid, that kind of scenario. That is what is called as ploidy. So the answer for this one will be, yeah, first option. First option would be correct for this. So the next question, lecithin, a small molecular weight organic compound found in living tissue. This is an example of phospholipids. So we have studied about this in the biomolecules chapter. So in alveoli, we do have lecithin, which is present in the phospholipid layer. Okay, next question. Match the following. So here, Clostridium botulinum, that will be responsible for formation of butyric acid. Saccharomyces cerevisiae, that will be responsible for formation of ethanol. And Trichoderma polysporum, that will be responsible for cyclosporin. And Streptococcus species, that will be forming streptokinase. So the option for this one will be 1. First option will be the answer. This is from biological classification, Monira chapter. Okay, okay, fine. Okay, next question ku polam ipo. Okay, so in this given figure, which component has thin outer wall and higher thickened inner wall? So in the picture pathina, so this is the guard cell. So guard cell ulla vandu, it will have a thick layer. Guard cell la ulla vandu thick layer irukum, velila vandu thin layer irukum. So guard cell is the one, idu vandu subsidiary cell. A vandu subsidiary cell. So in this, the guard cell is the one. So the option for this one would be C. Okay, C is the option. Guard cells is the option. So which of the following is an example of actinomorphic flower? Actinomorphic means when you are able to divide the flower into equal symmetry, then it is called as actinomorphic. We have seen about this zygomorphic. In zygomorphic, what will happen? We'll have the flowers where we'll be able to divide into 2 equal halves. So this will be called as zygomorphic, and this will be called as actinomorphic. So actinomorphic, Pisum, Sesbania, these all come under the Leguminosae family. Leguminosae family usually will be having the zygomorphic. So Datura is the one which will follow the actinomorphic family. So let's go to the next question. Okay, so a transcription unit in DNA is defined primarily by 3 regions in the DNA, and these are with respect to upstream and downstream end. So if you remember, we have studied about the transcription unit. So the transcription unit, obviously it should have a promoter. Promoter is the region where it will start, then it will have the structural gene, and then only it will have the terminator, the end part. So promoter is the region that the polymerase enzyme binds, and then it will start to, like we have studied about lac-operon, no? ZYA. So ZYA will be the structural gene, and finally the terminator will be there. So the answer for this one will be promoter, structural gene, and terminator. So the next question, auxin is used by gardeners to prepare weed-free lawns, but no damage is caused to grass, as auxin will not affect mature monocot plants. So what is the thing is, we have studied in auxin about 2,4-D and we have studied about 2,4,5-T. So these two, they are used as weedicides. They are used as weedicides. So normally வந்து ஒரு dicot agricultural farm இல்லனா ஒரு monocot agriculture farmனா ஒரு wheat மாதிரி அந்த மாதிரி farm எடுத்தோம்னா அந்த இடத்துல weeds வந்து என்னன்னா dicots இருக்கும். So அந்த இடத்துல when you spray excess of auxin, obviously they will inhibit, they will remove the weeds. So it will not affect the monocots, but it will affect the dicots in that region. So அதனால தான் அதை வந்து நம்ம weedicide-ஆ யூஸ் பண்றோம். This is from plant growth and regulation from that chapter. Okay. So this question, the cofactor of enzyme carboxypeptidase is zinc. This is a NCERT fact. The next question, the lactose present in the growth medium of bacteria is transported to the cell by action of permease. So we have studied about this lacopron. So lacopron, we have three structural genes, that is Z, Y and A. So they are responsible for one action. In this, Y is the one which is responsible for permease, and permease will allow the entry of lactose inside the cell. So the next question, which of the following can be explained on the basis of Mendel's law of dominance? Okay, so they are talking about the second law of dominance. Out of one pair of factor, one is dominant and the other is recessive. Yes, usually one is dominant and the other one is recessive. Allele do not show any expression and both characters appears as such in F2 generation. Alleles, they do show expression, like if it is dominant and recessive, in that scenario, dominant will be expressed. And factors occur in pairs in normal diploid plants. Yes, that is correct. Any diploid plant will have factors, that is alleles. Mendel used the word factors for alleles. And the discrete unit controlling a particular character is called as factor. That is correct. That is allele basically. And the expression of only one of the parental character is found in the monohybrid cross. That is, for example, when we do the capital T and cross it with small t, obviously one of the parent, though it is a heterozygous scenario, only one of the parent will be, we will be able to see. So that E is correct. Yes, D is also correct. Factors occur in pairs. That is also correct. Alleles show expression. So out of one pair of factor, one is dominant and the other is recessive. Yes, dominance is showed by that. So the answer for this one would be 4. Okay. So next question. So given below are two statements. Bt toxins are insect group specific and coded by gene CRY, LAC. And second statement, Bt toxins exist an inactive protoxin in B. thuringiensis. However, after ingestion by the insect, the inactive protoxin gets converted into active form due to acidic pH of insect gut. So the first statement, that is toxins are insect group specific and coded by gene, that is correct. The second statement, inactive protoxin in Bacillus thuringiensis, that is a wrong statement. So next question, given below are two statements. Statement 1, parenchyma is living but collenchyma is dead. That is a wrong statement. Parenchyma and collenchyma both are living. Only sclerenchyma is a dead tissue. Second, gymnosperms lack xylem vessels, but presence of xylem vessel is the characteristic of angiosperms. That is the correct statement. In angiosperms, usually you do have vessels, whereas in gymnosperms, you don't have vessels. So the first statement is wrong and the second statement is true. Okay, so the next question. Given below are two statements. Chromosomes become gradually visible under light microscope during leptotene stage. We have this meiosis. In meiosis, we have prophase 1. In prophase 1, we have 5 stages. So they are talking about the leptotene stage, the first stage. Usually it is visible during that stage. And statement 2, the beginning of diplotin stage is recognized by dissolution of synaptonemal complex. Yes, during diplotin, the fourth stage, the synaptonemal complex dissolution will be usually happening in this scenario. So, in the light of the above statement, choose the correct answer from the option. So, both the statements are correct. Okay, so as per meiosis, both the statements are correct. The first one is also correct and the second statement is also correct. Okay, so the next question, here we have to do the match. So nucleoli is the site for active ribosomal RNA synthesis, and centriole, it will be like a cartwheel, the axoneme arrangement, and leucoplast, this is for usually storing, and Golgi apparatus, that is formation of glycolipids. Okay, so nucleolus, the third option, and this is the second option. So the third option would be correct for this. Okay, so the list of endangered species was released by IUCN. Okay, so they are talking about the DNA which is present in the chloroplast. So we know the structure of chloroplast. So chloroplast usually it is a double membrane structure. It will have grana and we have these connections, and inside we have circular DNA. So the DNA which is present here, that is circular double stranded DNA which will be present. So that's why chloroplast and mitochondria, we have the concept called as endosymbiont and semi-autonomous. Okay, so next question. So which of the following are fused in somatic hybridization involving two varieties of plants? So somatic hybridization means when we take two cells, okay, usually for example, we have taken tomato and potato, and we use some chemicals and we fuse the nucleus. So this is what is called as somatic hybridization. So this is because of the protoplast fusion. Because of the protoplast fusion, this will be happening. So the next question, identify the correct description about the given figure. So cleistogamous flower showing autogamy. Cleisto means closed flower. This is not a closed flower. Well exposed anthers are there. Second question, compact inflorescence showing autogamy. This can be inflorescence, but it can't be autogamy because all are well exposed. So here, third option, wind pollinated plant inflorescence showing flowers with well exposed stamens. So this would be the correct statement because this is a wind pollinated flower where the stamens are well exposed. So the next question. So spraying sugarcane crop with which of the following plant growth regulator increase the length of the stem, thus increasing the yield? So increasing the internodal length, that is usually because of gibberellic acid. Increasing the height, that is strips, will be growing by auxin. Abscisic acid is an inhibitor and cytokinin will help in the lateral growth. So internode increase is because of gibberellic acid. Okay, so the next question, match the following. So Griffith worked on transformation experiment. Then Jacob and Monod, they proposed the semi-conservative model, and Gobind Khorana, he gave the genetic code, and Michelson and Stahl, Jacob and Monod. Okay, so in this, Khorana gave the genetic code, Jacob and Monod, Lacoperon, Griffith, transformation, Michelson and Stahl, semi-conservative. Okay, so the first one will be 3 and Jacob and Monod will be 4. So this will be the correct answer. Okay. So the next question, match the following. So GLUT4, that is for 4. Then insulin, that is a hormone, and trypsin is an enzyme, and collagen, that will be intercellular ground substance. So the correct option for this one would be 3. Okay, so the next question, in the C3 plants, some oxygen bind rubisco, hence carbon dioxide fixation is decreased. So the main difference between the C3 plant and C4 plant is the photorespiration concept. So photorespiration வந்து என்னன்னா, இப்போ நம்ம பிளான்ட்ஸ்ல யூசுவலா மார்னிங் டைம்ல it should undergo photosynthesis, the shoot part. ஆனா C3 plantsல it will undergo respiration, whereas in C4 plants that respiration will not happen. So C3 plants, some oxygen bind rubisco and hence carbon dioxide fixation is decreased. That is correct. In C4 plants, mesophyll cells sheath very little photorespiration, while bundle sheath cells do not show photorespiration. So this statement, the statement will be wrong. Okay, because obviously in C4 plants there is no photorespiration at all. So the first statement is true and the second statement is false. So this is the answer. Okay, the next question, identify the step in tricarboxylic acid cycle which does not involve oxidation of the substrate. So the Krebs cycle, so the first option, that succinyl coenzyme A to succinic acid, there is no conversion, there is no oxidation. We don't have NADH formation in this step, whereas all other steps we do have, like 4 steps, out of 4 steps, 3 steps we have NADH formation and 1 step we have FADH2 formation, whereas in this step there is no formation of NADH, instead GTP formation will be happening. Okay, so the next question, so where these reactions are going to happen. So citric acid cycle will be happening in the mitochondrial matrix. Okay, and glycolysis will be happening in the cytoplasm. Electron transport chain, that will be happening in the inner membrane, and proton gradient, proton gradient is high in intermembrane space. So when we take the mitochondria, we have the outer membrane, then we have the inner membrane. So this is the outer membrane, this is the inner membrane, this is the intermembrane space, and the center part, that is called as the matrix. So usually the matrix is the place where the Krebs cycle will be happening, and the electron transport will be happening in the that is the inner membrane, it will be happening. So the correct answer for this one will be option 4. Option 4 will be correct. So next question, which of the following statement is correct regarding the process of replication of E. coli. The DNA-dependent DNA polymerase catalyzes polymerization in 5' to 3' as well as 3' to 5'. This statement is wrong. Usually, the polymerization process starts from the 5' and ends at the 3'. And DNA-dependent DNA polymerase catalyzes the polymerization in the 5' to 3' direction. Yes, this statement is correct because this is replication. So, in replication, we have DNA-dependent DNA polymerase, whereas in transcription, we will be having DNA-dependent RNA polymerase. The next statement, the DNA-dependent DNA polymerase catalyzes polymerization in one direction, that is 3' to 5'. That is wrong because always the polymerization starts from 5' to 3' direction. And the DNA-dependent RNA polymerase catalyzes the polymerization in one direction, that is 5' to 3'. Here the word RNA is wrong because usually transcription happens because of RNA polymerase, but here we are talking about replication, so that's why that statement is also wrong. So the correct answer for this question will be the second option. Okay, the next question. In an ecosystem, if the net primary productivity of the first trophic level is 100x, what would be the GPP of the third trophic level of the same ecosystem? So they are giving the value here. So based on value, it will be the option 4. Okay, next question. Match the following. Choose the correct answer for the following options. So here, rose will be perigynous. And P will be marginal placentation. And cotton will be twisted aestivation. And mango will be droop. So for the correct answer for this one will be the third option. Okay, so for the next question, we have to choose the correct option again. Robert May, he was the person who proposed the global diversity. Robert May proposed the global diversity. Alexander von Humboldt, he was the person who proposed the species area relationship. Then Paul Ehrlich, he proposed the Rivert-Popper hypothesis. And David Tillman, he proposed long-term ecosystem experiments using outdoor plots. So for this, the correct option would be, okay. So the next question, we have to again do the match the following here. So mono Adelphus, we have studied about Adelphi in this morphology chapter. This is Adelphi is when the stamens are together. That is what is called as mono Adelphus condition. So here mono Adelphus is usually seen in China rose, that is hibiscus. Then diadelphous condition usually seen in pea plant, the 9+1 arrangement. 9 are together and 1 is separate. And I'm sorry, this is diadelphous. Diadelphous is usually in the pea plant. And polyadelphous condition, that is in citrus. And epiphyllous. Epiphyllous is when the tepal is attached to the stamen. Stamen is attached to the tepal. Because in lily we don't have petal, we have only tepal. So here we say it as epitepalous or epiphyllous condition. So based on this, the correct answer will be the third option will be the correct answer. Morphology chapter, again, this is. So the next question, read the following statement and choose the set of correct statements. So in the members of Phaeophyce, like in plant kingdom, we have studied about algae. We have green algae, brown algae, red algae. So chlorophyce, Phaeophyce, and rhodophyce. So here they are asking about the Phaeophyce, brown algae. So brown algae, asexual reproduction occurs usually by biflagellate zoospores. Yes, biflagellate pear-shaped gamete which will be present. That will be the condition. So A is correct. Then sexual reproduction is by oogamous method only. That is wrong. In case of red algae, there only we have only oogamy. In chlorophyce and Phaeophyce, you will have isogamy, anisogamy, and oogamy. All the three types of fertilizations are present here. So this is a wrong statement. And stored food is in the form of carbohydrates, which is mannitol or lamnarin. That is the correct statement. The major pigment are chlorophyll A, C, carotenoids, and xanthophyll. Here we'll have carotenoids, that is fucoxanthin, that is the one which gives the brown color. So yes, A, C is correct. Usually green algae will have A and B, brown algae will have A and C, and red algae will have A and E. E will be there. So this is brown algae. So A and D will be there in red algae. So A and C and xanthophyll. These are the two main pigments which give the brown color. And vegetative cells have a cellulosic wall, usually covered on the outside by gelatinous coating of algae. That is correct. The hydrocolloid material is present here. So the correct option for this will be A, C, D and A, C, D and E. Okay. So till this, we have finished the botany. Hopefully, all of you have done very well. Now, I'll pass on the session to the zoology faculty. Okay, and let's see few questions. Okay, 1 minute. Okay, we'll discuss the questions. Before that, I'll pass it to the zoology faculty. Thank you, guys.
Hello students, good evening. I hope எல்லாருமே NEET exam நல்லா பண்ணிருப்பீங்க and you are gonna be doctors. So உங்களோட first focus எப்பயுமே biology-ஆ தான் இருந்திருக்கும். And இப்போ வந்து next paper I'm going to discuss that is regarding zoology. So with no surprise, NTA has framed really good questions. So let's start now. So இப்போ first question பார்த்தீங்கன்னா first question match the following கொடுத்திருக்காங்க. So typhoid. So what is typhoid is caused by bacteria. இப்போ நம்ம கிட்ட A4 என்ன ஆப்ஷன் இருக்கு? Only one option. So இங்க பாத்தீங்கன்னா நம்ம வேற ஆப்ஷன் பார்க்கவே வேண்டாம். Only ஒரு ஆப்ஷன் தான் இருக்கு and quickly we will see A bacteria, that is typhoid will be caused by bacteria, leishmaniasis, protozoa, ringworm, fungus, filariasis, nematode. So இவ்ளோ ஈஸியான கொஸ்டின் அவங்க கொடுத்திருக்காங்க. நீங்க வேற ஆப்ஷன் பார்க்கவே வேண்டாம். Let's move on to the next question. So next question பாத்தீங்கன்னா it is from the chapter reproductive health and எல்லாருக்குமே தெரிஞ்சிருக்கும் 12th this is again an heavy weighted chapter. So இதுல பாத்தீங்கன்னா again the match the following non-medicated IUD. So non-medicated IUD na enna? It is Lippes loops. A is 3. So இங்க நம்ம கிட்ட ரெண்டு ஆப்ஷன் இருக்கு. Option 2 and option 3. So நம்ம next பார்ப்போம். So copper releasing IUD. So copper releasing IUD oda example enna? It is multi-load 375. So we have got this option. So you can see A is non-medicated IUD, which will be Lippes loop, and then you have copper releasing IUD, multi-load 375. Hormone releasing is levonorgestrel, which is LNG20, and implants progesterone. Now we'll move on to the next question. So next question on the pathina, it is statement based and it is from human reproduction chapter. Idla enna first statement enna sollirukanga, the presence of or absence of hymen is not a reliable indicator of virginity. Idu vande in the statement correct. Second statement என்ன சொல்லிருக்காங்க, hymen is torn during the first coitus only, which is wrong. எல்லாருக்கும் தெரியும், there will be given in the NCERT very clearly that hymen can be broken even during exercise, horse riding, or even when you are, lifting anything very heavy. So ultimately, in statement 2 is wrong. So statement 1 is true. So we are going with option number 1. So it's a very straightforward question they have asked. Coming to the next question. So again, this is regarding the case study. So in both sexes of cockroach, a pair of jointed filamentous structure called anal cerci are present. So உங்களுக்கு தெரியும் cockroachல common இருக்கிற structure என்ன, anal cerci. So that will be present on which segment? 10th segment. Again, a straightforward question from structural organization in animals chapter. Next, coming on to the next question. So இந்த தடவை பாத்தீங்கன்னா நிறைய match the following கொடுத்திருக்காங்க. The reason is they want to cover everything in one, question, all the concept. So இங்க பாத்தீங்கன்னா pons. நம்ம என்ன படிச்சிருக்கோம்? Ponsனா neuro, it is the one which is acting as a pneumosensitive center. But pneumotaxic center ni எதுவுமே இல்ல. So next we will see the hypothalamus part. So நம்மளுக்கு என்ன தெரியும்? Hypothalamus is a neurosecretory cell. So we will see B is 4. So where do I get that option? I get that option here. So this is how you should have solved the question without taking much time, but I'll explain this for you. So pons will be connecting the different region of brain. See, pons is again a part of the hindbrain. So it will be acting as nothing but the relay center. And then coming to the hypothalamus, which I have already told you that it is a neurosecretory cells. And why are we calling it so? Because it secretes the hormones for the posterior pituitary. And then coming to medulla, medulla is the one which will be controlling the process of the respiration and the gastric secretion. And cerebellum, of course, we are left with only one option, which provides additional spaces for neuron and so on. Coming to the next question. So which of the following is not a steroid hormone? So inga pathina options la, one second. So the first option on the progesterone, second is glucagon, third options on the cortisol nu kuduthirupanga. So please make a note of it. So idu cortisol la irukum. So progesterone, glucagon, third one the cortisol, fourth one the testosterone. So see, we are discussing the paper very early, so there will be some errors here and there. So I hope that you people will, overlook it. So idlande enna theriyuduna, glucagon mattum da proteinaceous, progesterone, cortisol, testosterone, idu ellame enna steroidal based hormone. So nammaloda answer enna, it is 2, which is glucagon. Now moving on to the next question. See, every year in the madri oru question avanga kuduthe thiruvanga, where they have asked which one is the correct product of the DNA dependent RNA polymerase to the given template. So given template in a TACAT. First I will solve it. So TAC na, here it will be A, then you will be getting U because it is an RNA. So instead of thymine, அங்க என்ன இருக்கும் base pair, it will be uracil. So AU, so G, UA, so I have got my answer, which is option number 3. Now coming to the next question. So next question, pathina, again a diagram based question directly taken from the diagram of the NCERT, where this is a skeletal structure and this is the smooth muscle and this is cardiac muscle. So let us see the options. So options la pathina skeletal romba kuduthirukanga. So my focus will be on the cardiac muscle. So cardiac muscle, heart, நம்ம கிட்ட ரெண்டு option இருக்கு. So we have to look at the one which is going best with it, and the skeletal muscle will be triceps, smooth is the stomach, and the cardiac is the heart. So ultimately the answer for this question is 4. Now coming to the next one. So this is again a question from the cell division, which is a fact-based question. So following are the stages of the cell division and என்ன கேக்குறாங்க stages correct orderல arrange பண்ண கேக்குறாங்க. So இங்க பாத்தீங்கன்னா first will be your gap 1 phase. So அது எங்க இருக்கு? So E I have got two options. So let me go to the next one. So next என்ன ஆகும்? Synthesis phase. So this is 1, 2. So E and then comes the C. So I have only one option left for that, which is option number 2. So the series goes like this: gap 1 phase, then you have synthesis phase, then you have gap 2 phase, then comes the karyokinesis followed by the cytokinesis. Now coming to the next question. So next question has come from the human health and disease chapter, again a 12th standard chapter. இதுல என்ன கேட்டிருக்காங்க, which of the following are autoimmune disorder? So autoimmune disorder, what happens? The cell starts attacking its own immune system. So here if you see, myasthenia gravis is an autoimmune disorder. I hope you know that, where antibodies start cleaving the receptors which are present on the sarcolemma. Then comes the rheumatoid arthritis, where there will be antibody starts accumulating in the cavity and it will form pannus granules. Now gout is because of the accumulation of oxalate salt. So இது வந்து autoimmune disorder கிடையாது. Then muscular dystrophy வந்து genetically, if there is any aberrations in the genes, that will cause the muscular dystrophy. So this is genetic disorder. Then this term may be new to you, but lupus is something that we usually come across. Commonly கேக்குற ஒரு term பாத்தீங்கன்னா lupus. So systemic lupus erythematosus அப்படின்னா என்ன அர்த்தம்? This will be affecting almost all parts of the body, right from any tissues of the body. So உங்களுக்கு எல்லாருக்குமே தெரியும், autoimmune disorder is because antibody starts attacking it, and in this case also antibody starts attacking many tissues of organs like even the brain, kidney, etc. So this may be a bit of a surprise for you, because in the statement, I mean in the disease வந்து நம்ம படிச்சிருக்க மாட்டோம், but the answer for this is A, B and E. So this is a little surprise which was given by the NTA, but yes, this is a common disorder. I hope all of you would have been known about it. Now coming to the next question. So next question again from biomolecules. This time biomolecules chapter நிறைய weightage கொடுத்திருக்காங்க. So let's see what they are trying to ask. So lipase எப்படி join பண்ணுவாங்க? With the lipids are joined by the ester bond. So A is 2. How many options do I have here? So I have only one option given here. So see, எனக்கு ஒரு time answer கிடைச்சிருச்சுனா, I am not even looking at the other option, and that's how you should have solved it. Okay, now coming here. So lipase is joined by the ester bond. Then the nucleases by the phosphodiester bond. Proteases, peptide bond, and amylase will be joined by the glycosidic bond. Amylase is nothing but a carbohydrate. So this is again a straightforward question. Now coming to the next question. So again, a direct question from the chapter of evolution, which all of you would have studied. Straightforward examples வந்து கொடுத்திருப்பாங்க. So the flippers of the penguins and dolphins are the example of what? Convergent evolution. So this is the answer. So coming to the next question. Okay, so again, one of the most expected question in NEET is to give regarding the respiratory volumes and the capacity. So இங்க என்ன கேட்டிருக்காங்கன்னா expiratory capacity. So when we talk about the expiratory capacity, that is a combination of ERV plus RV. So where do I get that option over here? Sorry, ERV and TV. So this is the option I have. A is 2, which is ERV plus TV, and functional residual capacity is nothing but ERV plus RV, and it goes on. So this is again a direct fact-based question, so I'm not discussing it much into detail. Now coming to the next question. So which of the following factors will not affect the Hardy-Weinberg equilibrium? Now again, this is from chapter Evolution, the 12th standard. And we all know when it comes to the postulates of the Hardy-Weinberg law, that says that the population is going to be constant. There should not be any kind of change in the frequency, etc. So, which one of the following factor will not affect the Hardy-Weinberg equilibrium? Apna, constant gene pool. So, you all know technically constant gene pool எப்பயுமே இருக்காது. But அதுதான் postulates Hardy-Weinberg lawல கேட்டிருக்காங்க. So, எந்த வந்து ஒரு condition வந்து இதை affect பண்ணாதுன்னா, that is constant. Coming to the next question. So below are some stages of the human evolution. Arrange them in the correct order of sequence from past to recent. So past in pathina. So first will be your Homo habilis, which will be so A followed by D, which is the Homo erectus. So where do I have that option? Yes. So where do I have that option? That is option number 2, which is A is Homo habilis, D is Homo erectus, C is Neanderthaliensis, and the last one will be nothing but us, which is Homo sapiens and sapiens. Now coming to the next question. So again, this is from the body fluids and its circulation chapter. So what they have asked, following are the stages of the pathway for conduction of an action potential through the heart. So I hope ellarku theriyu. Action potential engandu generate agum. It will be generated from SA node. So SA node landu thiruppi enga povum. It will be taken up by the AV node. So what is the thing which first I have got it is E and C. Inga pathina ore oru option da andha mari irukku, which is SA node, then AV node, AV bundles, followed by the bundle of branches, and lastly, Purkinje fiber. See, this is how, if you see, the paper has been set in a very easy manner. If you know even few information, you can easily go across with the option. Now coming to the next question. So, which of the following factors are favorable for the formation of oxyhemoglobin in alveoli? Again, a favorite NEET question, I will say, where they'll always talk about oxygen dissociation curve. So, என்ன favor பண்ணுது for the formation of oxyhemoglobin? So, oxyhemoglobin எப்போ form பண்ணும்? Whenever there is high partial pressure of oxygen. So, என்கிட்ட ரெண்டு option இருக்கு. So, definitely high PCO2, PO2 ரெண்டுமே ஒண்ணா இருக்காது. So, what am I left with? This one. So, it has to be high partial pressure of oxygen and lesser hydrogen ion concentration. Okay, next question. So, this is again the question which is coming from biotechnology chapter, biotechnology and its application. So, again, the match the following kuduthirukanga. If you see, this is something that I'm sure all of you would have done, well with. Alpha-1 antitrypsin. இது எதுக்காக use பண்றோம் for the treatment of emphysema. So let me see the options. A is 3 and cry1AB. இது வந்து is related. The cry gene is related with, it will affect which one? The corn borer. So ultimately, what is the answer that I'm left with? Okay, I'll discuss one more, which is cry1AC will be the cotton bollworm. And lastly, enzyme replacement therapy will go for the ADA deficiency. So the answer is 1. Okay, again, statement-based question from the human reproduction chapter. So inga vandu rendu statement kuduthirukanga, that is assertion and reasoning. Let's see what they are trying to say. So FSH acts upon the ovarian follicles in female and Leydig cells in male. So idhula what you have to understand is Leydig cells pathina, that will be under the control of what? LH. But inga enna kuduthirukanga that it is under the control of FSH itself, so that statement is wrong. So growing ovarian follicle secretes estrogen in female, where the interstitial cell secretes androgen in the male. So what we can go with this statement is correct. So A is false and R is true. Now coming to the next question, which is again from biotechnology and its principles chapter. So inga வந்து PBR 322 diagram கொடுத்திருக்காங்க. See, for a long period of time, diagrammatic based question in NEET கொடுக்கல. So in the time பாத்தீங்கன்னா, it's not a surprise. It was expected that அவங்க நிறைய diagram based questions கொடுப்பாங்க, and this is how they have framed the question this time. So though the question is very easy, இதுல என்ன கேட்டிருக்காங்க? X and Y they have marked. Apparama, they have asked what does it stand for. So ellarku theriyu X is nothing but the ORI site, ORI site, origin of replication. So gene X is responsible for controlling the copy number of linked DNA, and Y for the protein involved in the replication of plasmid, because Y is nothing but ROP. So idoda answer pathina it is 4. So again, the same, I don't know whether you'll be able to see the full option over here, but the option is 4. Again, as I've told you, due to the constraint of time, there will be some, errors here and there. I hope you can overlook it. So the answer for this question is going to be option number 4. Okay, next. Again, a straightforward question, which is from the 12th chapter, human health and disease or a drug and abuse portion landa vandirukku. So cocaine, so you can see the cocaine will be extracted from the erythroxylum, and the heroin will be from Papaver somniferum. So we are going with option number 2. Again, this is a very simple question, a fact-based question. Now coming to the next one. Fine. So it is from animal kingdom part. And it says annelids are true coelomates, which is correct. Poriferans, see, poriferans have nothing to do with the coelom. They are like the basic organisms which will be falling under the phylum Porifera. And Ascalminthes are pseudocoelomates. Again, this is wrong. And Platyhelminthes are true coeloms. So what are we left with? It is option number 4. Now coming to the next question. So this is again from your 11th standard excretory system chapter, which says, in the nephron, the descending limb of loop of Henle is impermeable, but you know it is permeable to water. So inge statement on the wrong side. So we will not even proceed further because we know that is wrong. Now statement 2, PCT is lined by simple cuboidal, simple columnar. So ultimately both the statements are wrong. So you have to go with option number 4, that says both statement 1 and 2 are false. Okay, now coming to the next one. So the fibrous joints, again, a favorite NEET question. Every year they will touch this. So what does it, what are they asking? Of course, they match the following. Fibrous joints on the skull do not allow any movement. So if you see, this is again so simple. I'm just left with, if you see, there is only one option. A3 வந்து வேற ஆப்ஷனே இல்ல. So நீங்க வேற ஆப்ஷனை பார்க்கவும் வேண்டாம். But okay, I'll explain this. We'll quickly see. Cartilaginous joints is 1. Then hinge joints is the knee. Ball and socket joint is nothing but the humerus and the pectoral girdle. Now coming to the next question. Which of the following is not a natural traditional contraceptive method? So except Waltz, because this is a barrier based method, all other are gonna be natural and traditional contraceptive method. Again, the chapter from which we, say it is heavy weightage and you can expect a lot of questions, though it's very simple from reproductive health. Now coming to the next question. Animal kingdom question straightforward கேட்டிருக்காங்க. The first is pleurobrachia. So pleurobrachia பாத்தீங்கன்னா, the example is going to be for ctenophora. So A2. So there are two options that I am left with. So B is radula. So the radula will be for mollusca. So B is 1. So I am left with only one option, which is option number 4. So கொஞ்சம் இன்ஃபர்மேஷன் தெரிஞ்சா கூட நீங்க அழகா இந்த கொஸ்டினை சால்வ் பண்ணிருக்கலாம். ஸோ பார்த்தீங்கன்னா pleurobrachia is for ctenophora, radula is for mollusca, stomachord பார்த்தீங்கன்னா for hemichordata, and lastly air bladder for osteichthyes. Now coming to the next question. So this is again, they have asked about what are the things that you can go across again from the cell chapter. So axoneme ne vandana, first what should strike you is it is present in the cilia and flagella. So A is 2. So I have two options here. So I'll go across for another option. Cartwheel pattern pathina enga nama paakalam centriolar. So it is B is 1. So A2, B1, C4, D3. So this is a very straightforward question. Crista will be found in where? Mitochondria. Satellites will be found on the chromosome. So I need not explain much on this. It's a fact-based question. Now moving on to the next question. So it says, which of the following statement is incorrect are used to produce a small scale bacterial culture? So inga enna question ketirukanga? Small scale. So bioreactors. So first option பாத்தீங்கன்னா என்ன சொல்லிருப்பாங்க? Bioreactors are used to produce small scale bacterial culture. But உங்களுக்கு எல்லாருக்குமே தெரியும் bioreactors is for large scale, production of microorganisms. To be very specific, the bacteria. So which statement is wrong? Option 1 is incorrect, because they have said it is used small scale production and all we will do easily in the lab itself. So we need not require, bioreactors for that. So ultimately the answer is 1. Now coming to the next one. So this is again a basic which we expect all of you to know about it. So idu enna kekranga, various phases of prophase 1 they have given. So diakinesis is completion of terminalization of chiasmata, and B is chitin, which is appearance of the recombination nodules. So idhula namukku enna option kedaikudhu? A is 2, B is 4, C is 1, and D is 3. So this is again a fact-based question. I'm not getting into the detail of it. Now coming to the next one. So you can see the paper, you may feel it is easy, but it is lengthy because they have given a lot of statement and match the following. But if you are very clever enough to just see one option and mark it, you could have done it within the fixed time. So common cold is caused by rhinovirus. So A is 2. I will not even go further. And I go option which is A2. Hemozoin, hemozoin will be released from where? After the plasmodium infection, it will be released from the erythrocytes, which is responsible for recurrent chills and fever. So you can see B is 1, which is plasmodium, and hence the answer will be 1. I'm sorry, here there is again a printing mistake, which has to be 3. So we have to go with option number 1. So common cold is rhinovirus, which is option number 3. Hemozoin is plasmodium. Vidal test is for the typhoid, and allergy is for dust mites. I hope all of you will be having the paper. So ungalku and prachana varakudadu. But while solving, yes, because it was given as 2. So you please make a note Options are not going to be wrong. So the option is 1. So again, kindly overlook the typing mistakes. Now coming to the next one. So they have again given an assertion and reasoning type of a question which says breastfeeding during the initial period of infant growth is recommended by the doctors for bringing a healthy baby, which is completely correct. WHO recommends that all the females should feed the baby for 6 months minimum, and it depends upon the individual how long they want to go. Colostrum contains several antibodies, absolutely essential to develop the resistance for the newborn baby. Correct. உங்களுக்கு எல்லாருக்குமே தெரியும் newborn babyக்கு immunity ரொம்ப கம்மியா இருக்கும். So அந்த immunity first எங்கிருந்து கிடைக்கும்? From the colostrum, which will be having the antibody IgA. So both the statements are correct, and the answer is 3. So it is the correct as well as the correct explanation of assertion. So we are going with option number 3. Coming to the next one. So again, pathina match the following kutirkanga. So pterophyllum. So pterophyllum is an angelfish. So A is 3. So in the A3 and okay, I have got 2 options for this. B pathina Maxine is hagfish. We have given the same thing. C pathina Pristis, which is the sawfish. So I will be going with option number 4. Again, a direct straightforward question. நீங்க NCERT படிச்சிருந்தீங்கன்னா அதுல எல்லாத்துக்குமே இந்த examples கொடுத்திருப்பாங்க. Now coming to the next one. So here the question says the TI plasmid of Agrobacterium tumefaciens stands for. Very simple question. TI எல்லாருக்குமே தெரியும். It is nothing but tumor inducing plasmid. So இதோட option என்ன? Answer is 1. Now, next question, which of the following is not component of fallopian tube? So, the component of fallopian tube are infundibulum, ampulla, and isthmus, and they themselves have given uterine fundus. So, it is a part of uterus. So, what is the answer here? It is 3, because it is not a component of fallopian tube. More than this, an easy question cannot be framed where they have given the answer within the option. Okay, coming to the next question. So next question is regarding the chromosomal disorders. So Down syndrome pathina, that is because of the chromosomal abnormality in chromosome 21st. So A is 3. Yeah, again, I need not even look into other option, which is the first option. And because of the direct questions, I'm not going to discuss into it. The answer for this question is going to be 1. Coming to the next question. Fine. So again, animal kingdom la the question ketirukanga. The following are the statements about the non-chordates. So first thing, chapter when we talk about the non-chordates and chordates and the characteristics the papo. So let's see which and all are the characteristics which are about the non-chordates. So the question is about the non-chordates. So the pharynx is perforated by the gill slits. Notochord is absent, so this is not a character. CNS is dorsal, they have written. Heart is dorsal if present, which is correct. Now, post-anal tail is absent because that will be found in the chordates. So, what is the answer that we are left with? Again, a fact-based question, direct answer, B, D, and E. So, again, from the evolution where they have given the geological time scale, Mesozoic era. So, when we talk about the Mesozoic era, that is related to birds and reptiles. Proterozoic era is lower invertebrates. Now, let me see the option. A is 3, B is 1. Okay, I have to go one more. So, Cenozoic era is for mammals, C is 4. So, I have got my answer, and D is going to be fishes and amphibians, Paleozoic. So, the answer is option number 2. Now, coming to the next statement, which says, the cerebral hemisphere are connected by the nerve tract known as the corpus callosum, which is correct. Again, a fact-based question. The brainstem consists of the medulla oblongata, pons, and what they have given here, cerebrum. It has to be cerebellum. So ultimately, the statement is wrong. So what are we left with? Statement 1 is correct. Statement 2 is incorrect. Now let us see the next question. So they are saying identify the correct option of A, B, C, D with respect to spermatogenesis. So LH, so you see A, they have not given anything, but what is the GnRH will be having its effect on what? LH as well as FSH. So my A is going to be FSH. So where do I have that? So I have got two options here where they have given FSH, and let me just go to the next one. So B is LH. Which cell will secrete androgen, that is testosterone? You all know it is the Leydig cells. So I've got my answer, which is option number 2. So let us quickly see this of a flow chart. So FSH, then comes the Leydig cell, which will secrete the androgen and it will help in the formation of the spermatids. And C is the Sertoli cells, which will be providing the required nourishment and the factors. So see the answer, though I have told it is 3. So FSH, then comes the Leydig cell, the Sertoli cells, and lastly it will be spermiogenesis. Now coming to the next one. Again, it is a straightforward question where RNA polymerase 2 is related with snRNAs and tRNA. So A is 4 and I have got only one option. And because it is a fact-based question, I'm not going to discuss it further. So you can see that the answer is option number 2. Now coming to the next one. So this is again, this time they have given a lot of importance for the disorders. So exophthalmic goiter. So exophthalmic goiter is caused due to the hypersecretion of the thyroid hormone, and what is the symptom that we see over there is the protruding eyeballs. So A is 3. Again, I have got two options here, so I will see further. Acromegaly, which is caused due to the hypersecretion of growth hormone after puberty. So if you see, that is again 4. So I have got my answer here, which is A3, B4. So let us see quickly. Cushing syndrome, it is related to the cortisol, and the cretinism, you know, it is caused by the hyposecretion of thyroid gland, specifically during the condition of pregnancy. So the answer is going to be option number 2. Okay, so talking about again the match the following. Unicellular gland epithelium. So unicellular, you know, except goblet cells, all other are going to be the multicellular gland. So A is 3. So if you see, I have only one option here. So I'm not even further looking up at other things. And because it's a straightforward question, I don't think there's much to discuss over here. So the answer for this is option number 1. Okay, so again a statement-based question which says, bone marrow is the main lymphoid organ where all blood cells, including the lymphocytes, are produced, which is correct. Both bone marrow and the thymus provide the microenvironment for the development and the maturation of T lymphocytes. In the end statement to me correct. Bone marrow and thymus the primary lymphoid organ, and they will be doing all this function. So ultimately, nammaloda answer enna, both statement 1 and 2 are correct. Now, let's talk about the next question. So, either again case study landa vandirukku, which is the cockroach. So, the structure used for storing of food nale, it has to be what? The crop. So, A is 4. So, I have two options here. So, I will just go further. So the ring of 6 to 8 blind tubules at the junction of foregut and midgut is nothing but the gastric CK. So B is 2. So A, the structure used for storing of crop is 4 and B is 2, C is 3 and D is 1. So it is going to be option number 3. Now the next one. So again, a statement-based question they have given here. So the question says, choose the correct statement given below regarding the juxtamedullary nephron. So you know that juxtamedullary nephron enna pannu, that will go deep. The loop of Henle will go deep into the medulla. So you have to keep that in mind. So let's see the options now. So the loop of Henle of juxtamedullary nephron are first option itself is correct, which says loop of Henle of juxtamedullary nephron runs deep into the medulla. If you see other things, juxtamedullary nephron outnumber the cortical. This is wrong. You know cortical nephrons are maximum. 80% of it will be cortical nephron. Types of nephron are two, cortical and juxtamedullary. So which is more in number? That is cortical. But இவங்க என்ன சொல்றாங்க, juxtamedullary nephron outnumbers the cortical. So in the statement வந்து wrong. Next, juxtamedullary nephron are located in columns of Bertini, which is again, columns of Bertini is nothing but the extended part of enna cortex. So அதனால இந்த option வந்து wrong. Fourth என்ன சொல்லிருக்காங்க, renal corpuscles of the juxtamedullary nephron lies in the portion of renal medulla. So this is again, they are talking in the terms of renal corpuscle, which has nothing to do with that of the juxtamedullary nephron. So in this statement again will not be applicable. So what are we left with? Option number 1. Now coming to the next question. So it says, again we have to do match the following. So P wave. P wave is nothing but depolarization of atria. P is 3. So A is 3. See again, now further options paaka vendam. They have, there is only one option which says that P wave is what? Depolarization of atria. Either again straightforward based, fact based question. So further discuss panama. I'll just tell you the answer, which is for 196, the answer is going to be option 4. Now coming to the next one. So either one, it is related to the blood group. இங்க என்ன சொல்லிருக்காங்க, as per the ABO blood grouping system, the blood group of father is B plus, mother is also A plus, child is O plus. So you imagine father B plus irukanga and mother is A plus, but the child has turned out to be O plus. இதுல இருந்து என்ன தெரியுது, ரெண்டுமே heterozygous condition இருக்கு. So that means because O is a homozygous recessive condition. So ரெண்டு அலீலுமே homozygous recessive இருந்தா தான் O கிடைக்கும். So இதுல இருந்து நம்மளுக்கு what we can go across with. So all the 3 alleles will be present because IA, IO, here, sorry, IB, here IA, IO. So you will have the allele for B as well as A as well as for O. So what is the option I could see? Yes, it is option number A. I mean, this is the A and the answer is going to be option 3. Okay, now coming to, again, this is a straightforward fact-based question. So we will just see and look into the statement, which says Gauss's competitive exclusion principle states that the two closely related species competing for different resources cannot exist indefinitely, which is of course false. Now, according to the Gauss's principle, during the competition, the inferior will be eliminated. This may be true if the resources are limiting, and this is true. So ultimately, we are going with option number 2, which says statement 1 is false, but statement 2 is true. Because it's a direct question, so I hope you can understand that. Yeah, we'll go on to the next question. So the next question says, regarding the catalytic cycle of an enzyme action, select the correct sequence step. So in that, the first part will be always an enzyme will bind to the substrate, correct? So what will be the first thing is substrate. So I'll mark it in the number. So one is going to be nothing but substrate binding to the active site. Then there will be formation of substrate enzyme complex. So let me just check the option. So A, so E, A. So I have got my answer here. So let us see whether this fits in or not. So as I've told you, first will be the binding, then the complex formation. D is chemical bonds of substrate being broken, and then we will be going with the remaining options. So it is E, A, D, C, and B. So it is option number 3. Now coming to the last question of zoology part, which says mitochondria and the chloroplast are both double membrane bound organelles, which is correct. And statement 2 says the inner membrane of the mitochondria is relatively less permeable as compared to the chloroplast, which is incorrect. So ultimately, we are going with, again, this is a fact-based and straightforward question. So the answer is statement 1 is correct, but statement 2 is incorrect. So children, stay tuned. We will be showing the answer key for biology, that is both botany and zoology, within few minutes. And after that buffer time, we will start with the discussion of chemistry question. So thank you so much for joining, and I hope ellarume nalla pannirpeenga and you will all get into medical colleges. Thank you so much for joining in.
Like say, come back to like say chemistry paper analysis. பார்த்தீங்கன்னா வந்து கெமிஸ்ட்ரி எல்லாமே என்சிஆர்டிலேருந்து டைரெக்டாக கேட்டிருக்காங்க. ஃபிசிக்கல் கெமிஸ்ட்ரி வந்து லைக் சே கேல்குலேஷனாக இந்த வாட்டி ரொம்ப ஈஸியாக கேட்டிருக்காங்க. ஸோ ஐ திங்க் லைக் நீங்கள் எல்லாருமே நல்லா பண்ணியிருப்பீங்கன்னு நினைக்கிறேன். ஸோ லைக் வி வில் ஸ்டார்ட் த டிஸ்கஷன். லைக் சே லெட்ஸ் மூவ் டு ஃபர்ஸ்ட் கொஸ்டின். ஸோ ஃபர்ஸ்ட் கொஸ்டின், இஃப் யூ சி லைக் it is like say from redox as well as electrochemistry mixture. So actually if you see like say H2 is converted into O2. So first I need to predict the oxidation state. So if I see the oxidation state of this oxygen, this will be -2. And we all know that oxidation state of elementary form will be 0. Therefore, this will be 0. Therefore, like here, if you see the change, 2 electrons. And we very well know that Faraday என்றது a charge carried by 1 mole of electron. அப்ப இங்க 2 electron இருந்தா எவ்வளவு Faraday வரும்? 2 Faraday. So like say A answer வந்து 2 Faraday. Okay, next option B பாத்தீங்கன்னா 1 mole of MnO4 minus is converted into Mn2 plus கொடுத்திருக்காங்க. So like first I have to find the oxidation state. MnO4 minus, it is converted into Mn plus 2. So இதோட oxidation state of Mn is like say plus 7. அப்போ இதுல எவ்வளவு சேஞ்ச் இருக்குன்னு பாத்தீங்கன்னா like say 5 electron. Therefore this will be equal to 5 Faraday. So 5 Faraday corresponds to option number 4. So இப்போ வந்து பாத்தீங்கன்னா like say A corresponds to like say 2. So I can easily eliminate option 2 and 4. ஏன்னா A first optionலயும் third optionலயும் மட்டும்தான் A2 இருக்கு. Okay, next B வந்து ஆக்சுவலா வந்து நான் சால்வ் பண்ணிட்டேன். B வந்து எவ்வளவு வந்திருக்குன்னா 4 வந்திருக்கு. So I can go with the answer 3. No need to solve like the other things. Okay. So which reaction is not a redox reaction? So we all know that redox reaction, it will have oxidation as well as like the reduction will happen simultaneously. Okay, if you see this option number 2, let's check whether there is a change in oxidation state or not. So இதோட oxidation state எவ்வளவுன்னு பாத்தீங்கன்னா +2. So chlorine ஓட oxidation state எவ்வளவுன்னு பாத்தீங்கன்னா -1. Again Na ஓட oxidation state பாத்தீங்கன்னா +1 and sulfate ஓட oxidation state பாத்தீங்கன்னா -2. Again let's check like say barium ஓட oxidation state எவ்வளவு +2. Sulfate ஓட oxidation state again -2. Na ஓட oxidation state +1. Cl ஓட oxidation state -1. அப்போ கொடுத்திருக்கிற ஆப்ஷன்ல பாத்தீங்கன்னா only in option number 2 like the oxidation state change ஆகல. So this is not a redox reaction. So answer is 2. Okay. 53rd question, intramolecular hydrogen bonding is present in. So first of all, what is intramolecular hydrogen bonding? So intramolecular hydrogen bonding அப்படின்னா see, it has to take place within the same molecule. Then that is called as intramolecular hydrogen bonding. Intermolecular ரெண்டு மாலிகுலுக்கு நடுப்புற நடந்தா அந்த ஹைட்ரோஜன் பாண்டிங் பேரு இன்டர்மாலிகுலர் ஹைட்ரோஜன் பாண்டிங். சோ இப்போ ஃபர்ஸ்ட் ஆப்ஷன் பாத்தீங்கன்னா இது வந்து இன்டர்மாலிகுலர் ஹைட்ரோஜன் பாண்டிங். சோ இரண்டு மாலிகுலுக்கு நடுப்புற இந்த ஹைட்ரோஜன் பாண்டிங் நடக்கும். Okay, if you see second option, again this is also intermolecular hydrogen bonding. This is also what? Intermolecular hydrogen bonding, which is happening between, like, say, two molecules like this. And if you see the third option, this so basically here, like, say, the hydrogen bonding is happening within the molecule. Therefore, this is a classical example of intramolecular hydrogen bonding. Let's go to fourth option and check. Again, this is going to be intermolecular hydrogen bonding because it will happen between two species or two molecules. Therefore, answer will be 3. Okay, next question. This question straight away from NCERT textbook and Pelling solution. Like, say, what is Pelling solution A they are asking. So basically Pelling solution it is made by mixing Fellings A plus Fellings B. So basically what is Fellings A? So Fellings A is aqueous copper sulfate. This is the straight line from NCERT textbook. And Fellings B is sodium potassium tartarate. So, like, say, this is not Fellings A, actually this is Fellings B. So aqueous sodium citrate is not the correct answer. So aqueous copper sulfate is the correct answer. Okay, coming back to next question, they are asking 1 gram of sodium hydroxide was treated with 25 ml of 0.75 molar HCl solution. The mass of sodium hydroxide left unreacted is equal to. So initially they have given, like, say, 1 gram of sodium hydroxide. So it was treated with 25 ml of 0.75 molar HCl solution. So they are asking, we need to find the mass of sodium hydroxide left unreacted. And they are given it as mg. So I am converting this, therefore it will be 1000 mg. So simple formula, like, say, I can apply equivalence concept. So gram equivalence of NaOH will be equal to gram equivalence of HCl. So it will be W I need to find because I need to find the mass of NaOH left unreacted. So W by the molar mass of NaOH is 40. Okay, N factor is 1. And again, gram equivalence of HCl, they have given, like say, volume 25 ml, then molarity, they have given us 0.75 molar. So it will be 25 multiplied by 0.75. So ml I am converting into step. So I am dividing by 1000 into 1. If I solve this, I will be getting the weight of sodium hydroxide. So like actually the answer is 250 mg. If you solve this, like you will be getting and subtract it and you will be getting the answer is 250 mg. Okay, next a straightforward question from chemical bonding, and let's say chemical bonding has lot of weightage in every NEET paper. This time also like we got one question. So like the first option NH3. So the steric number is basically 3 plus 1. That is, like if you see the structure of ammonia, it has 3 sigma bond plus 1 lone pair. That's why I have written 3 plus 1, which is equal to 4. So steric number 4 corresponds to sp3. Okay, so sp3, and then if you see the geometry of this, it is trigonal pyramidal. So A goes with the option number 1. So I can do easy option elimination because no other option corresponds. A is equal to 1. So I will go with option number 3. That is A1, B4, C2 and D3. Okay, next question is the NEE0 value for M13 plus M12 plus couple is more positive than that of Cr3 plus slash Cr2 plus or Fe3 plus slash Fe2 plus is due to the change of. Okay, let's see. Like they are telling M13 plus slash M12 plus, this couple has more positive SRP value. More positive SRP value compared to Cr3 plus slash Cr2 plus or Fe3 plus slash Fe2 plus, and they are asking it is due to the change of. So let's write the electronic configuration of M13 plus. So this will be 3D4. So M13 plus will be changed to M12 plus. Therefore, electronic configuration will be 3D5 and it is off filled, and the stability of M12 plus is more than M13 plus. That's why it has a good reduction tendency. That's why it has positive SRP value. Therefore, the electronic configuration is changing from D4 configuration to D5 configuration. So, like say option number 1 says D4 to D5. That is the correct answer. Again, a question from thermodynamics. This time, lot of question came from thermodynamics. So, isothermal. Iso means constant. Thermal means temperature. So, like say carried out at constant temperature A. Let's check the option. So, again A1 and then here A is 4 is given, A is 2 is given. So, I will go with this. Let's say B also isochoric is basically like iso constant, choric means volume. So, carried out at like say constant volume, again B3. So, like say option number 2, straightforward question from thermodynamics. Okay, next question. Activation energy of any chemical reaction, that is EA of any chemical reaction, can be calculated if one knows the value of. See, we can't calculate the activation energy by knowing the orientation of reactant molecule. So basically, orientation of the reactant molecule during collision will make sure, like say, this is process is happening. So rate constant at two different temperatures. So we very well know this equation: log K2 by K1 is equal to EA by 2.303 or 1 by T1 minus 1 by T2. So by knowing the rate constant at two different temperature only, we can calculate the activation energy. Therefore, answer is 2. Then, like say, they are given a compound with a molecular formula C6H14 has two tertiary carbons. Two tertiary carbon. Let's try like option number 1. So it has dimethyl butane. So then option number 2, it has 2, 2 dimethyl butane. Option number 3 is hexane and 2 methyl pentane. Now, if you see, this is basically called as tertiary carbon. This is basically called as tertiary carbon. So they are asking two tertiary carbon present in a molecular formula C6H14. So first option le rendu tertiary carbon irukku. In second option, if you see, like say I don't have any tertiary carbon, 0 tertiary carbon. Third option, again, like say no tertiary carbon, therefore again, like say I can say 0 tertiary carbon. Okay, fourth option, again, like say I have only 1 tertiary carbon. Therefore, the answer is 1. Okay, then they are asking spin only magnetic moment is same for which of the following ions. So basically, the number of unpaired electrons should be same. The number of unpaired electrons should be same. Then my value of mu will be same. Now let's check. Option A is given as titanium 3 plus. Option B is given as Cr2 plus. Option C is given as M12 plus. Option D is given as Fe2 plus. Option E is given as Se3 plus. Now I need to write the electronic configuration. So basically, like this will be 3D2 4s2. And this will be 3D5 4s2. Like say this is titanium and this is electronic configuration of chromium and manganese. Sorry, this is 3D5 4s1. So this will be 3D5 4s2 and this will be 3D1 4s2. So this is the configuration of titanium, this is the configuration of chromium, this is the configuration of manganese, and this is the configuration of scandium. Now from this 3 electrons have been removed. Therefore, this goes to 3D1 and from this 2 electron is removed. This goes to 3D4 and from this again 2 electrons are removed. Therefore, 3D5 and again like say here 3 electrons are removed. So 0. And then Fe2 plus again this is 3D6 4s2, therefore 3D6. Therefore, here, like say option A, number of unpaired electron, that will be 1. Option B, number of unpaired electron will be n is equal to 4. Option C, number of unpaired electron n is equal to 5. Option D, again, number of unpaired electron will be 4. Option E, number of unpaired electron will be 0. So only B and D have same number of unpaired electron. Therefore, only B and D will have same spin only magnetic moment. So option is 3. Okay, next question. Arrange the following elements in increasing order of electronegativity value. So we very well know that according to Pauling scale of electronegativity. I know the value of fluorine is 4. Then oxygen is 3.5. Then I have nitrogen, which is 3.0. Then they have given carbon, which is 2.55. Then, like say, they have given silicon, which is like say 1.9. So basically, like say fluorine, then oxygen, then nitrogen, then carbon, then silicon. Option 3 is correct. Which one of the following alcohols reacts instantaneously with Lucas reagent? So they are asking, edu vandu instant Lucas reagent oda react agum. So first of all, what is Lucas reagent? So that is basically ZnCl2 plus. This is called as Lucas reagent. Okay, இது என்ன வந்து பண்ணும் அப்படின்னா, it will differentiate primary, secondary and tertiary alcohol. Tertiary alcohol இதுல Lucas reagent போட்டா instant turbidity கிடைக்கும். Okay, secondary alcohol Lucas reagent add பண்ணா turbidity will appear after 5 minutes. Primary alcohol when I add Lucas reagent, room temperature turbidity form agad. So this is the key concept to solve this question. So first option is not a tertiary alcohol. So second option, if you see this OH group is attached to the 3-degree carbon. So this is tertiary alcohol. And again, this OH is attached to the primary carbon. Therefore, it doesn't give any turbidity. And again, this is going to be like secondary alcohol. It will give turbidity after 5 minutes. ஆனா அவங்க என்ன கேட்டிருக்காங்க, இன்ஸ்டன்டேனியட்டா யார் டர்பிடிட்டி கொடுப்பாங்க? So answer is 2. Okay, next question. This question is from coordination compounds. So given below are two statements. Statement 1, both CONS363 plus and COF63 minus complexes are octahedral but differ in their magnetic behavior. Yes, coordination number of CONS363 plus is basically 6. And again, coordination number of COF63 minus, again this is 6. If coordination number 6 means I will get the geometry octahedral. So complexes are octahedral. This is correct. But differ in their magnetic behavior. So I have to analyze the nature of the ligand in this case. Let's say in compound 1, NH3 is a strong field ligand. Compound 2, F is a weak field ligand. So because of this, I'll be getting different configuration. Therefore, it will have differ in the magnetic behavior. Therefore, statement 1 is correct. Statement 2, CONS363 plus is diamagnetic, whereas COF63 minus is paramagnetic. In the light of the above statement, choose the correct answer from the option given below. So let's check, like say NS3. So basically, like it is going to be strong field ligand. Therefore, like force pairing will take place. So like say I will be getting a diamagnetic over here, and like say here no force pairing will take place. Therefore, paramagnetic in nature. This is also correct. So like say given options are, so both statement 1 and statement 2 are correct. So I think option number 3 is the right answer. Given below are two statements. Statement 1, they are giving the boiling point of hydrates of group 16. Elements follow the order. The order is given as H2O greater than H2TE greater than H2SC greater than H2S. And this is a direct question from NCERT. And we very well know that boiling point is directly proportional to van der Waals. But the order is different because H2O will give intensive hydrogen bonding. Because of this, this order is also different. So obviously, statement 1 is correct. This is a fact-based question from NCERT. Statement 2, on the basis of molecular mass, H2O is expected to have lower boiling point than the other members of the group, but due to the presence of extensive hydrogen bonding, this is very, very important. Due to the presence of extensive hydrogen bonding, it has higher boiling point. This is also correct. So I think like options are given as statement 1 is true, but statement 2 is false. No. And statement 1 is false, but statement 2 is true. No. So statement 1 and statement 2 are true. So answer is 3. And again, this is the question from atomic structure, quantum numbers, very easy. So basically ML. So it represents what? So basically ML represents orientation of orbital. So I think like I'll be getting like say first option as well as like this fourth option. MS represent orientation of spin of electron. So again, like say here also 4, here also 4. So I can't eliminate it. So like I have to go with another thing. So like L will be like a shape of orbital. So that will be 1. So I will go with option number. And again, this is the question from organic chemistry. Like say they are given option A is basically a double bond is given. And then I am getting 2 moles of a cyclic ketone. And this is basically ozonolysis process. Therefore, the reagent required for this is like say fourth one. So like say A option number 4, the reagent is O3/Z10H2. Next, B, they are given benzene. And this has been converted into so basically like this is said to be a Friedel-Crafts alkylation process, and here something is missing. Actually, I have to get AlCl3. So basically like this is an electrophilic substitution reaction. So B matches with 1. Now we will go for option elimination. So like here also A is 4 and like say here also A is 4. So I can easily eliminate option number 2 and option number 4 directly. Now, like say I know B also 1, here also B is 1, here also B is 1. Therefore, again, like I have to go for one more option. And again, you see this alcohol group has been converted into ketone. And obviously, like say this is due to this CrO3. And then like say like C is 2 here. So I answer is 1. And again, like say here an alkene is given and then they are converting into aldehyde. So CH2 single bond CH double bond CH2. Now like say first reagent if I put again like say this is hydrolysis. So I won't get like say because it is in primary position, so I won't get a primary alcohol. Again here I won't get a primary alcohol. Only in option 1 and option 4 there is a possibility I will get a primary alcohol. So like say I can add BH3 and then like say second reagent will be H2O2/OH minus. So this H2O2 indicates it follows anti-Markovnikov rule. So basically I have to add water across this double bond. So according to anti-Markovnikov rule, therefore I will get CH2 CH2 single bond CH2OH. Okay, next, like say this OH has been converted. This group has been converted to aldehyde. Therefore, mild reducing agent is like mild oxidizing agent. Mild oxidizing agent is PCC. Therefore, if I am going to use PCC, Therefore, I will get this compound. So answer is 4. The reagent with which glucose do not react to give the corresponding test. Obviously, glucose will react with tolerance, and then it will react with NH2OH, it will react with HCl. It don't react with skiffs as well as like NaHSO3. This is again a direct question from NCERT biomolecules. So the answer is B and E, option number 1. Then ethane, again, like say it contains 1 sigma bond, therefore A corresponds to 3. So again, I can do option elimination, therefore 3 and 4 are eliminated. So I have 1 and 2. Then ethene, again, I have like say 1 sigma bond and 1 pi bond. Again, B is 4, here also B is 4. Then again, carbon molecule slash O2, then like say ethine is given. So ethine has like say 1 sigma bond and 2 pi bond. So like say D is 1. So actually, like say option number is 1. So ethane contain, so basically 1 sigma bond. Then like say ethene, it contain 1 sigma bond and 1 pi bond. Ethine contain 1 sigma bond and 2 pi bond. Therefore, like option number 1 is the correct answer. Among group 16 element, which one does not show -2 oxidation state? So they are given tellurium, oxygen, polonium, and then like say selenium. So oxygen, sulfur, selenium, tellurium, polonium. When I am moving from top to bottom of this group, metallic character increases. Therefore, metal do not show negative oxidation state. Out of this, like say polonium is said to be like say higher metallic character. Therefore, polonium do not show -2 oxidation state. Therefore, answer is 2. For the reaction, 2A gives B plus C. A Kc value is given as 4 into 10 to the power -3. At a given time, the composition of the reaction mixture A is equal to B is equal to C, 2 into 10 to the power -3 molar. They are given. So they are given some reaction. So 2A gives B plus C. Okay, fine. Then they have given Kc value for this equation is 4 into 10 to the power -3. Okay, fine. At a given time, like say the composition of the reaction mixture, the concentration of A as well as B as well as C, it is given as 2 into 10 to the power -3 molar. So then which of the following is correct? So they are telling reaction has a tendency to go in backward direction, option number 1. So option number 2, they are telling reaction has gone to completion in forward direction. Option number 3, reaction is at equilibrium. So basically, like in order to do this question, first of all, I need to predict QC. So QC I can derive from this equation. Therefore, it will be concentration of B into concentration of C divided by concentration of A. So concentration of B is given in the question as 2 into 10 to the power -3 molar. Again, C is also 2 into 10 to the power -3 molar divided by. Again, concentration of A is also 2 into 10 to the power -3 molar power 2. If I cancel this, I will get QC will be 1. Therefore, QC is greater than KC. Therefore, the reaction will move in backward direction. Oh, like say reaction has a tendency to go in backward directions. Option number B is correct. Which plot of ln K versus 1 by T is consistent with Arrhenius equation? So we know Arrhenius equation, that is K is equal to A e to the power minus EA by RT. So if I multiply by ln and all, I will get ln K is equal to ln A minus EA by R times 1 by T. So I can write it as equal Y is equal to C minus MX. So in x-axis, I am plotting 1 by T. So the slope will be negative. So x-axis, I am plotting 1 by T. So this is said to be the negative slope. So the slope will be equal to minus EA by R. So this is an intercept which is nothing but ln A and y-axis ln K. So option number 2 is correct for this question. In which of the following equilibrium Kp and Kc are not equal? So it is very simple. So first of all, so delta Ng, if it is going to be 0, then Kp will be equal to Kc. If delta Ng is not equal to 0, then Kp is not equal to Kc. Now, how do I find delta Ng? So delta Ng is basically total number of moles of gaseous products minus total number of moles of gaseous reactants. Okay, let's check one by one option. So first option, so in product side, again I have delta Ng, total number of moles of gaseous product is 2 minus, and again reactant side, again 1 mole, so 2, it will be 0. So first option, obviously Kp will be equal to Kc. They are asking not equal, therefore this is not the correct answer. Second, again if I am going to calculate delta Ng value, and again this is gas, so 1 plus 1 is 2. So 2 minus 2 is equal to 0. Again here Kp is equal to Kc. Therefore, this is not the correct answer. Option number 3, delta Ng is equal to total number of moles of gaseous product. 1 plus 1 will be 2 minus, like say this will be 1. Therefore, 1. So delta Ng is not equal to 0. Therefore, in this equation, my Kp is not equal to Kc. Therefore, correct answer is option number 3. Given below are two statements. The boiling point of 3 isomeric pentanes follows the order of N-pentane, isopentane, neopentane. This is correct because we very well know that boiling point is inversely proportional to branching. So here branching takes place, and again more number of branching takes place, therefore boiling point decreases. So this is statement 1 is correct. A statement 2, when branching increases, the molecule attains a sphere. So in case of neopentane, if you see branching increases, this molecules attains a spherical form. Okay, so this results in smaller surface area for contact, and due to which the intermolecular forces between the spherical molecule are weak, thereby lowering the boiling point. So this is also correct. So the option is, so statement 1 is correct, but statement 2 is incorrect. This is not the correct answer. The statement 1 is incorrect, but statement 2 is correct. This is again not the correct answer. So both statement 1 and statement 2 are correct. So this is the correct answer. We all know that, like say, boiling point decreases on increasing branching. So both statement 1 and statement 2 is correct with respect to this theory. So I will go with answer number 3. The compound that will undergo SN1 reaction with a faster rate. We very well know that a rate of SN1 will be directly proportional to the stability of carbocation. And obviously, tertiary carbocation is more stable than 2 degree than 1 degree. So if I see over here, this is going to be my tertiary carbocation. So like say option 2 is correct. The energy of an electron in the ground state for helium ion is minus x joules. Then that for an electron in N is equal to state for Be3 plus ion in joules. So basically E1 of He plus divided by E2 of Be3 plus is equal to, you very well know that E is directly proportional to Z square by N square, and this is negative. So I can say that 2 square by So it will get cancelled. So I'll be getting 1, that is -1. So like say not minus 4x, not minus 4 by 9x. So E1 of HE plus will be equal to E2 of BE3 plus, that will be equal to -1. So I will go with option number 3, that is -x. In which of the following process entropy increases? So option A liquid evaporates to water. So if liquid is converted into gas, obviously, like say my entropy will increase because randomness in gas molecule is more, obviously entropy will increase. So this is obviously entropy will increase. When temperature of a crystalline solid lowered, whenever temperature is lowered, obviously my entropy will also be lowered. So in option number B, like say my entropy won't be increased. Option C, like say again, I will calculate my delta Ng. So delta Ng, obviously this is gas, and this is no gas. So obviously my delta Ng will be 2. And obviously when delta Ng increases, my delta S will also increases. Entropy will increase. So option C, entropy will increase. So option D also, like say again, 2Cl gas is there. So my delta Ng value will be 1. And again my like entropy increases. So in D also my entropy increases. So the correct answer is A, C, D, option number 1. On heating, some solid substance change from solid to vapor state without passing through liquid state. The technique used for the purification, it's just a 9th standard question. Actually, solid is converted into gaseous state by bypassing, like say, formation of liquid state. This process is basically called as sublimation. It's just a fact-based question, so direct question, and again, like say, the answer is 4. Match list 1 with list 2. So this is a good question actually from coordination compound. This is isomerism. So option A, CO, NH3 5, NO2, Cl2. So this NO2 can bind in 2 ways to the central metal atom. Like say it will bind through oxygen or it will bind through nitrogen. Therefore, this is basically called as a linkage isomerism. Okay, let's check the other option. I can eliminate 1 and 4. So NH3 5 SO4 Br. So basically this will exchange and this will exchange. This is ionization isomerism. So obviously B will be 3. So A will be 2, B will be 3. And this is exchange of ligand between the coordination sphere. Therefore, this will be like say coordination isomerism. So A is 2. So like option 1 can be eliminated, option 4 can be eliminated. So I will end up with option number 2 and option number 3. So again, A is 2 in both the cases. So I will go with B. So B is basically like a ionization isomerism. So answer 3 is correct. Given below are 2 statements. Statement 1, aniline does not undergo Friedel-Crafts alkylation reaction. Okay, so aniline, when I treated with AlCl3, this is basically an Lewis acid. This is basically Lewis base. So this will donate the electron. It will form a complex. Therefore, like it do not undergo Friedel-Crafts alkylation reaction. Statement 1 is correct. Statement 2, aniline cannot be prepared through Gabriel synthesis. So it is basically Gabriel synthesis. So we will be taking phthalimide. So basically, like say I have to react this with chlorobenzene. So where SN2 reaction is not possible, therefore this attack is not possible. Therefore, aniline cannot be prepared through Gabriel synthesis. This is also correct. So in the light of the above statement, choose the correct answer from the option given below. Statement 1 is correct, but statement 2 is false. This is wrong. Statement 1 is incorrect, but statement 2 is true. This is wrong. Both statement 1 and 2 are correct. So this is correct. So this question answer is 3. Arrange the following elements in increasing order of first ionization enthalpy. So obviously beryllium will have, like say, completely filled configuration. It will switch it. So obviously I will be getting lithium, then this is lithium actually, then boron, then beryllium, then carbon, then nitrogen. Answer 4 is correct. This is a fact-based question from periodic table. The highest number of helium atom is sent. So 4 gram of helium, option number 1, they are given 4 gram of helium. So basically, like if I am going to find the mole, so basically I will be getting 2 Ne. So here, like say I will be getting second option 2.27 liter by 22.7. So again, like say I'll be getting 0.1. So third option 4 mole. So 4 into Na. So that will be 4 into 6.022 into 10 to the power 23. So this will have highest number of helium atoms. So answer is 3. The most stable carbocation among the following is. So I need to check the number of alpha hydrogen. In option 1, this is alpha position. Second, this is alpha, this is alpha, this is alpha. Third one, again, this is alpha, this is alpha. Fourth one, again, this is alpha, this is alpha. So in second option, like say alpha hydrogen is more, therefore stability will be more. Therefore, the most stable carbocation among the following is answer number 2. The Henry's law constant KH values of 3 gases A, B, C in water are. So they are given A as 145 and B is 2 into 10 to the power minus 5. They are given in kilobar. This is also in kilobar. And C will be 35 kilobar. So basically, we very well know that KH is inversely proportional to solubility. So obviously here A will be less soluble, B will be more soluble, C. So obviously answer is, sorry, B will be greater than 4 only. Next, a compound X contains 32% of A, 20% of B, and remaining percentage of C. Then the empirical formula. So basically, what is empirical formula? Empirical formula is nothing but the simplest ratio of a substance. So they have given atomic mass of A is 64, B is 40, C is 32. So like say A, I can say that 32 by 64. B will be 20 by 40 and rest will be C. Will be 48 by 32. So this will be 3 by 2 and this will be 1 by 2 and this will be 1 by 2. So I'll get A 1 by 2, B 1 by 2, C 3 by 2. The answer is A, B, C 3. Okay, the products A and B obtained in the following reaction respectively are. This is like say question directly from NCERT. It is a preparation of alkyl halide. So A will be H3PO3 and B will be POCl3. So option number 2 is the answer. This is a fact-based question from NCERT. The plot of osmotic pressure pi versus concentration. For a solution, give a straight line with the slope 25.73 liter bar mole inverse. The temperature at which the osmotic pressure measurement is done. They are asking temperature. So we very well know that osmotic pressure formula pi is equal to CRT. They are plotting the graph between osmotic pressure and concentration. That means they are plotting the graph between pi versus C. And the slope will be basically RT. That value is given as 25.73. And they are asking us to find what is the temperature. So we very well know that, like say, T will be 25.73 divided by R value is given in the question. So 0.083 liter bar mole inverse per Kelvin. If I am going to, like say, do the calculation, I will be getting 310 Kelvin. So it will be 37 degree Celsius. So option 3 is correct. Again, for the given reaction, let's say some alkene is given and this is said to be a strongest oxidizing agent. And we very well know that the alkene will be oxidized to acid. So this is not an acid, this is not an acid, this is option number 2. And this is not an acid aldehyde. So option number 4 is the correct answer because it will oxidize to carboxylic acid, 2 moles of carboxylic acid. Option number 4 is correct. Okay, now here given below are two statements. Statement 1 is CONS36 is a homoleptic complex, where CONS34Cl2 is a heteroleptic complex. What is basically homoleptic complex? A complex with one kind of ligand. One kind of ligand, they are called a homoleptic complex. Now, obviously, this has only one kind of ligand. So it is homoleptic complex. This is correct. Then what do you mean by heteroleptic complex? Complex with more than one kind or type of ligand, then that is heteroleptic complex. Here it has two different types of ligand. This is heteroleptic complex. Therefore, statement 1 is correct. Statement 2, complex NONS36 has only one kind of ligands. It is correct. But CONS34Cl2 has more than one kind of ligands. It is also correct. Therefore, in the light of the above statement, choose the correct answer from the options given below. Statement 1 is true, but statement 2 is false. This is wrong. Statement 1 is false, but statement 2 is true. This is again wrong. Both statement 1 and statement 2 are true. This is correct. So option number 3. During the preparation of more salt solution, ferrous ammonium sulfate, which of the following acid is added to prevent hydrolysis of Fe2 plus ion? This is a direct question from salt qualitative analysis. So answer is dilute sulfuric acid. Identify the correct answer. Dipole moment of NF3 is greater than that of NS3. This is wrong. It is like say lesser. 3 canonical forms can be drawn for carbonate ion. So C double bond O minus O minus. So if I am going to do resonance, so 3 canonical forms can be drawn for CO3 2 minus. This is the correct answer. Given below are certain cations. Again, like say this question is from salt analysis. Using inorganic qualitative from 0 to 6. So we have like say group 0 to 6. And then Cu2 plus, that is B belongs to group 2. This is again fact-based question. Then Al3 plus belongs to group 3. So again, like say I can easily eliminate like say 1 and 2. So B, A, B, C. So I will go with B, A, and this belongs to fourth group. This belongs to second group. This belongs to third group cation. This belongs to fifth group cation, and this belongs to sixth group cation. So the correct answer is 3. Again, identify the major product C formed in the following reaction sequence. So they are given CH3, CH2, CH2I. This on treatment with NaCN. Cn minus is a strong nucleophile. Therefore, SN2 mechanism takes place. So this will have delta positive, this will have delta negative. Therefore, this will go and attack this. And I minus is a very good leaving group, leaves the substrate. So as a result, I will get CH3, CH2, CH2, Cn. This is my A. So A on treatment with OH minus partial hydrolysis. We very well know that Cn on complete hydrolysis, it will be converted into carboxylic acid. So Cn on partial hydrolysis, it will be converted into amide. So basically CH3, CH2, CH2, C double bond O, NH2 I will get. This is my B. B on treatment with NaOH plus Br2. This we very well know. It is Hoffman bromamide reaction. So basically this group has been replaced with amine. So it will give a propyl amine. So answer is 3. The rate of reaction quadruples and temperature changes from 27 degrees Celsius to 57 degrees Celsius. Calculate the energy of activation. Again, there is okay. So our value is given as 8.314 joules per Kelvin per mole. Log 4 value is given as 0.602. So we very well know that log K2 by K1 will be equal to EA by 2.303 R 1 by T1 minus 1 by T2. So T1 is basically given as 27 degrees Celsius and T2 is given as 57 degrees Celsius. You convert this into Kelvin and you substitute over here. And like the rate of reaction quadruple, so this will become log 4. So log 4 is equal to EA by 2.303. So R value is given in the question as 8.314. And I'll be getting 1 by 300 minus 1 by 330. If I solve this, I will get the value of EA, that is 38.04 kilojoules per mole. Consider the following reaction in a sealed vessel. And we very well know that in closed vessel only equilibrium will take place. With a concentration of N2 is given as 3.0 into 10 to the power minus 3 molar. O2 is given as 4.2 into 10 to the power minus 3 molar. And NO is given as 2.8 into 10 to the power minus 3 molar. So this is basically an equilibrium arrow. So 2NO gas gives N2 gas plus O2 gas. If 0.1 mole inverse of NO is taken in a closed vessel, what will be degree of dissociation alpha of NO at equilibrium? Now we very well know that, like say they are given this equation as 2NO gas gives like N2 gas plus O2 gas. Now the concentration of N2 is given in the question as 3.0 into 10 to the power minus 3. O2 concentration is given as 4.2 into 10 to the power minus 3 molar. Then NO concentration is given as 2.8 into 10 to the power minus 3 molar. So from this equation, I can write the expression of Kc. So that will be concentration of N2 multiplied by concentration of O2 divided by concentration of NO raised to the power 2. If I substitute the respective concentration in this equation, I will get Kc is equal to 3.0 into 10 to the power minus 3 molar multiplied by, so O2 will be 4.2 into 10 to the power minus 3 molar divided by NO will be 2.8 into 10 to the power minus 3 molar power 2. If I solve this, I will get Kc value as 1.6. When they are asking alpha over here. Therefore, like 2NO gas gives N2 gas plus O2 gas. At time t is equal to 0, they are telling 0.1. Okay, is taken in a closed vessel. What will be degree of distillation? This will be 0.1 minus 0.1 alpha. This will be 0.1 alpha by 2. This will be 0.1 alpha by 2. Therefore, Kc will be again equal to 0.1 alpha by 2 into 0.1 alpha by 2 divided by 0.1 minus 0.1 alpha part 2. And Kc value we already found it, that is 1.6. If I solve it, I will get alpha value as 0.717. The work done during reversible isothermal expansion of 1 mole of hydrogen gas. So mole is given as 1. Then 25 degrees Celsius. So temperature is given as 25 degrees Celsius. Then pressure P1 is given as 20 atm. Then P2 is given as, like say, 10 atm. So like you know the formula. So work done during irreversible isothermal expansion is minus 2.303 nRT log P1 by P2. If you substitute the respective values over here, you will be getting the answer as mass in gram of copper deposited by passing 9.6487 ampere. So current is given as 9.6487 ampere through a voltmeter containing copper sulfate solution for 100 seconds. So time is basically given as 100 seconds. So I need to find mass in grams of copper. So we very well know that W is equal to E by F into Q. So W is equal to, so E very well know that equivalent weight. So molar mass is given as 63 by N factor is 2. So like 63 by 2 divided by a Faraday's constant is given in the question as, so 96,487 multiplied by, this will be 9.6487 multiplied by 100. If I solve this, I'll get answer 4. Major products A and B formed in the following reaction sequence are. So again, this question is from haloalkanes and haloarenes. So I have OH. If I treat this with PBr3 and again SN2 mechanism will take place, this OH will be substituted by Br. So I will get A as this one. So from this, I can eliminate option number 2. Okay, then alcoholic KOH. A is treated with alcoholic KOH. And we very well know that alcoholic KOH, the mechanism is E2, which is also called as beta elimination. There are two possibilities. So this is basically alpha. So this is also basically alpha. So they are asking major product Therefore, if I remove this, and obviously like I'll get a pi bond and then Br will go away. So A will be this and B will be this. So option number 3 is correct. The pair of lanthanide ions which are diamagnetic is, we very well know that it is again fact based question. Ce4 plus will have 4f0 and Yb2 plus we have 4f14. So both have, like say, no electron here, like say all are paired state. Therefore, diamagnetic is Ce4 plus and Yb. Okay. Okay, like say 63 question, there is some error, I guess. Like say we will have a different PPT for that. Actually, there was an error in option 1. Like say initially it was given as double bond. Like say here it is single bond. Again, which one of the following alcohols reacts instantaneously with Lucas reagent? So we very well know that tertiary alcohol will give instantaneous turbidity. So there was error in first question. Like say initially they have given double bond over here. Like say now they have changed it to single bond. That's it. Again, option number 2, because this is only tertiary in nature, it will give you instant turbidity. Okay, thank you so much for watching this video. And then after some buffer time, we will be having physics session. Thanks a lot.
Fine. So very good evening, students. So we are almost to the last subject today. So let us quickly get into the paper discussion of physics part of your NEET 2024. Overall paper pathina, it's easy to moderate. There are only few questions that are tricky. Most of the questions, if you are good in basics, then you can easily crack it. So let's quickly get into the first question. So here is question number 1. So a tightly wound 100 turns. So turns is involved here of radius 10 centimeter coil. So basically it's a circular coil. Current value is given as 7 ampere. Magnitude of the magnetic field at the center. So what is the formula for magnitude of magnetic field at the center due to circular coil? You have turns here, but turns vandita are mu naught Ni by 2R, where mu naught value they have given us. N value is 100, I value 7 and kurdachi. R value is 10 centimeter. Please be careful. Centimeter to meter conversion panano. So once you do and then you solve it, then you will get option 1 as the answer. That is 4.4 millitesla. Just substitute very simple calculation. You will get as pi ki badala 22 by 7. It will be even easier. That 7 and all will get cancelled. So what you get is 4.4 millitesla. Right. So let us move on to the next question. So again a theory question. Again in the paper la nariya theory question vandirukku. Kittathata around 15 questions are theory based. So if you are good in theory part, again nallave in the paper la score pannikalam. Fine. So inga varrappa idu magnetism la vandirukku. So they have given different magnetic materials and they are asking about the susceptibility. Right. So அப்போ ஒரு diamagnetic அப்படின்னு பாக்குறப்ப one of the important characteristics of diamagnetic என்னது? It has an negative susceptibility. So அங்கே negative susceptibility அப்படின்றப்ப you'll have the negative. So A should have 2. So there are only 2 options. நீங்க ஹாப்பியா 1 and 2 eliminate பண்ணிடலாம். Now ferromagnetic. Ferromagnetic are strongly magnetic. Strongly magnetic என்ன அர்த்தம் அதுக்கு? susceptibility ரொம்ப அதிகமா இருக்கும்னு அர்த்தம். So B is 3. நீங்க இந்த ரெண்டு பார்த்தோம்னா முடிச்சுட்டு போயிருக்கணும். If you are very smart in this, then only this option matches. So if, for clarification, paramagnetic will have lesser susceptibility, non-magnetic will have zero susceptibility. So obviously the option matches C4 and D1. So option 3 in this case would be right. So next question, again question பார்க்கிறதுக்கு ரொம்ப பெருசா graph value எல்லாம் கொடுத்து உங்களை பயமுறுத்தி வச்சிருக்காங்க. But it's very simple. What they are asking, work done along path BC. They are not asking work done of the total process. Only BC மட்டும் தான் கேக்குறாங்க. இந்த BC என்ன process? இந்த BC பாத்தீங்கன்னா எப்படி இருக்கு? Volume constant இருக்கு. When volume is constant, it's an isochoric process. So this BC is isochoric. Isochoric அப்படின்னு கண்டுபிடிச்சிட்டீங்கன்னா work done என்ன? நேரா நீங்க எந்த கால்குலேஷனும் பண்ண வேணாம், it is 0 because work done in isochoric is 0. You don't have to touch your pen. If you find BC is isochoric, then work done 0 போட்டு போயிட்டே இருக்கலாம். You don't have to do any maths involved there. So these kind of questions are there. அவங்கள அப்படியே பயமுறுத்துற மாதிரி பயமுறுத்தி, but questions are really simple there. So let's move on to the next question now, the fourth one. So again another theory question. An unpolarized light strikes a glass surface at Brewster's angle. Brewster's angle na என்ன அர்த்தம்? போலரைஸ் ஆகுதுன்னு அர்த்தம். அப்ப Brewster's angle இன்சிடென்ட் ஆச்சுன்னா எது போலரைஸ் ஆகும்? ஸோ அப்போ இன் ஜென்ரல் ஒரு இன்டர்ஃபேஸ்ல லைட் இன்சிடென்ட் ஆச்சுன்னா ரிஃப்ளெக்ட் ஆகும், ரீஃப்ராக்டும் ஆகும். பட் த கண்டிஷன் ஃபார் Brewster's angle என்னது? Brewster's angle கரெக்டா இன்சிடென்ட் ஆச்சுன்னா ரீஃப்ளெக்டட் கண்டிப்பா கம்ப்ளீட்டா போலரைஸ் ஆயிடும். ஸோ ரீஃப்ளெக்டட் லைட் வில் பி கம்ப்ளீட்லி போலரைஸ். பட் ரீஃப்ராக்டட் கம்ப்ளீட் போலரைசேஷன் நடக்கணும்னு அவசியம் கிடையாது. So which means refracted light will be only partially polarized. So but the correct answer would be the reflected light is completely polarized and refracted is partially polarized is the perfect answer for it. So now let us move on to the next question. Again a very good question. Just நல்ல trick வச்சிருக்காங்க இதுல. So ideal transformer சொல்லிட்டாங்க அவங்க VSVP. So ideal transformer க்கு என்ன இது? NP by NS is VP by VS. Right, your voltage is directly proportional to number of turns. ஆனா क्वेश्चनல என்ன ட்விஸ்ட் வச்சிருக்காங்க? VS by VP ன்னு கேக்குறாங்க, VP by VS. சோ அப்ப NP by NS என்ன கொடுத்துறாங்க நமக்கு? 1:2. ஆனா கேக்குறது என்ன? VS by VP. சோ இதுல இருந்து என்ன சொல்லலாம்? VS by VP is nothing but 2:1. So option 4 is going to be the right answer. So very very tricky question. நீங்க கொஞ்சம் அப்படியே டைரக்ட் ரேஷியோ அப்படி பார்த்து 1:1 போட்டீங்கன்னா போச்சு. கேர்ஃபுல்லா இருங்க. இங்க கேக்குறது VS by VP. That's the catch in this question. So let us move on to the next question. And logic gate question. ரெண்டு logic gate question வந்திருக்கு. So if those are good in logic gates, you have a bumper prize with you. So logic circuit provides the output Y as per the following truth table. எதுவும் பெருசா பார்க்காதீங்க, அந்த B and Y மட்டும் பாருங்க. B 0 இருந்தா Y 1, B 1 இருந்தா 0. அப்ப 0 1 1 0. அப்ப என்ன சொல்லலாம்? Y ன்றது என்ன? It is the not of B. So B bar will be your right answer. You don't have to check. அழகா இது ரெண்டு மட்டும் பாத்தீங்கனாலே தெரிஞ்சிடும். 0 1 1 0 0 1 1 0 அவ்வளவுதான். அப்ப இதோட பார் தான் Y. அப்ப Y is nothing but B bar. So option 1 is the right answer. ஹாப்பியா போட்டு போயிட்டே இருக்கலாம். நெக்ஸ்ட் கொஸ்டின். திஸ் இஸ் ட்ரிக்கி. இது நீங்க மெஷர்மெண்ட்ஸ் படிச்சிருந்தாதான் இது போட முடியும். ஸோ வெர்னியர் கான்ஸ்டன்ட் அப்படின்னு சொல்லிருந்தாங்கன்னா அது லீஸ்ட் கவுண்ட்னு அர்த்தம். வெர்னியர் கான்ஸ்டன்ட் சொல்லலாம், லீஸ்ட் கவுண்ட் அப்படின்னு சொல்லலாம். So அப்ப your vernier constant or least count is nothing but 1 MSD minus 1 VSD. So this is the base idea. If you're not knowing this, then nothing can be done. But this is a very simple. Now what data they have given? N plus 1 vernier scale division. So N plus 1 VSD is coincide with N divisions of main scale. So it will be NMSD. Right. So now go. இதுல இருந்து 1 VSD, how can I write? 1 VSD is nothing but N by N plus 1 MSD. So go substitute in this. What you will get is 1 by N plus 1 MSD அப்படின்னு வரும். 1 MSD என்ன கொடுத்திருக்காங்க? 0.1 millimeter. ஆனா ஆன்சர் எதுல கேக்குறாங்க? சென்டிமீட்டர். So again conversion உங்களுக்கு பிடிச்சிருக்காங்க. When you convert, then obviously you'll get, when you substitute 0.1 millimeter and convert to centimeter, it will be 1 by 100. So 1 by 100 into N plus 1 will be the right answer. In this is a very good question. One of the top in the slot liye konja tricky question appdin paakrappa indha question sollalam. So let us move on to the next question now. So eighth question, the maximum elongation. So elongation appdina enna kandupidikanum? First del L kandupidikanum of a steel wire of length 1 meter. So L is given, The elastic limit அப்படின்றப்ப stress பத்தி பேசுறாங்க. நான் thought you can get from the unit. The elastic limit அப்படின்னு சொல்றப்ப they are talking about the breaking stress over there, and Young's modulus is given. So we know Young's modulus is F into L by A into del L. So இதுல del L தான் கேக்குறாங்க. So del L கேக்குறாங்கன்னா del L is nothing but F into L by A into pi. F by A கொடுத்தாச்சு, that is your elastic limit. F L கொடுத்தாச்சு, that is 1 meter. Young's modulus கொடுத்தாச்சு, 2 into 10 to the power 11. Substitute and simplify, you will get option 3 as the answer, that is 4 millimeter. So again, if you are good in manipulating the formula, it's an easy question for you. Let us move on to the next question now. Yes, again a good question from NLM, a very basic question. Horizontal force of 10 Newton is applied to the block A as shown. Mass of block A and B are 2 and 3. இந்த மாதிரி மல்டிபிள் பிளாக் வந்துட்டாலே ஃபர்ஸ்ட் அது ஒரு சிஸ்டமா எடுத்துக்கோங்க. when you consider as a system, then என்ன சொல்லலாம், ஒரு 5 kg ஆயிடுவது, அதுல 10 நியூட்டன் ஃபோர்ஸ் ஆக்ட் ஆகுது. அப்ப this 10 F is equal to ma தானே, அப்ப 10 is equal to 5 into A. அப்ப A is nothing but 2 meter per second square. Your step 1 is done. Then they are asking the force exerted by block A on B. அப்ப B மட்டும் எடுங்க. B ல என்ன, இந்த 10 நியூட்டனுக்கு B க்கு சம்பந்தமே கிடையாது. இந்த 10 நியூட்டனை தூக்கிட்டு போய் நீங்க B ல சப்ஸ்டிட்யூட் பண்ணவே கூடாது. அப்ப B ல யார் ஆக்ட் பண்ணுவா, வெறும் the contact force between A and B தான். அப்ப அதுதான் நம்மள கேக்குறாங்க, கரெக்ட்டா? FAB தான் கேக்குறாங்க. அப்ப only force acting is that. அப்ப இதோட ஆக்சலரேஷன் நம்ம கண்டுபிடிக்கலாம். as a system it has an acceleration of 2. again Newton's law, FAB is nothing but mass of B. What is mass of B is given 3. A என்ன 2. so FAB is nothing but 6 Newton. So FAB in this case is going to be 6 Newton. So option 1 again. Those are good in NLM. It's an easy question to take 4 marks. Let us move on to the next question quickly. Okay, so another theory question from wave optics. If the monochromatic source in Young's double slit experiment is replaced by white light, so in general your bandwidth, everything is lambda d by d. Your fringe width in the first position or n lambda d by d, n lambda d by d. அதெல்லாமே everything is based on lambda. So the point here is, I'm not going into it. The point here is everything, every parameter in Young's double slit is going to be based on is lambda. So அப்படி பார்க்கறப்ப all fringes of equal fringe length is தப்பு, because lambda கேத்த மாதிரி உங்களுக்கு fringe இது மாறும். Interference pattern will disappear அப்படின்னு ஒன்றும் அவசியம் இல்லை. White light is nothing but combination of 7 colors. அது disappear ஆகிறதுக்கு ஒன்றும் அவசியம் இல்லை. There will be central dark fringe, there will be central. So இது ரெண்டுத்துக்கு தான் உங்களுக்கு confusion. So idea is, when you have a double slit experiment, when you have a light over here at the central thing, the path difference will be 0. When you consider central fringe from both the sources, your path difference is basically 0. So if path difference is 0, then it will be a constructive interference. அப்ப இந்த இடத்துல 7 color, all which are will form its central bright fringe at the central fringe. அப்ப 7 கலரும் ஒண்ணா தெரிஞ்சா நம்மளுக்கு எப்படி தெரியும், white. அப்ப there will be a central bright fringe surrounded by, then அதுக்கு அடுத்து lambda கேத்த மாதிரி உங்களுக்கு position of different colors will change. So option 1 in this case is the right answer. A good question again though. Now next question, if you are good in interpreting graph, then again but a very good question, graph interpret பண்ண தெரிஞ்சதுனா சூப்பரா மார்க் எடுக்கலாம். These are the questions that makes the difference between your 160, 170 and 140. Okay, a graph shows variation of 1 by lambda square and its kinetic energy E. So about what we know, lambda de Broglie wavelength அப்படின்னு வந்துட்டா, what we know about it, lambda is equal to, இங்க எனர்ஜி பத்தி பேசிருக்காங்க. அப்ப choose a formula that has an energy. Be smart in choosing the formula. H by root of 2me. ஆனா இப்ப நம்மளுக்கு என்ன வேணும், 1 by lambda square வேணும், E வேணும். அது ரெண்டு ரிலேட் பண்ணும். அப்ப lambda square வேணும்னா first ஸ்கொயர் பண்ணுங்க. ஸ்கொயர் பண்ணீங்கன்னா lambda square is equal to h squared by 2me. இப்போ 1 by lambda square வேணும். அப்போ 1 by lambda square வேணும் அப்படின்னா 1 by lambda square is 2m by h squared into E. ஸோ அப்போ அவங்க 1 by lambda squareக்கும் ஈக்கும் தான் கிராஃப் கேட்குறாங்க. they are asking graph not by lambda and E, lambda square ஈக்கு கேட்கல, 1 by lambda squareக்கும் ஈக்கும் தான் கிராஃப் கேட்குறாங்க. அப்போ 1 by lambda square E, இது ஒரு y அப்படின்னு எடுத்துக்கலாம். இது எல்லாமே கான்ஸ்டன்ட், அது ஒரு m. அப்போ இது xன்னு எடுத்துக்கிட்டா, this is in the format of y is equal to mx. So y is equal to mx format இருந்தா கிராஃப் எப்படி இருக்கும், it will be a straight line graph. So option 2 is going to be the correct answer. Again, a very good question to test your basics. Now let us move on to the next question. Fine. In the following circuit, the equivalent capacitance between terminal A and B. So, நீங்க Wheatstone bridge அப்படின்னு படிச்சிருப்பீங்க. அது ரெசிஸ்டர்ஸ்க்கு மட்டும் இல்ல கெபாசிட்டர்ஸ்க்குமே அது அப்ளிகபிள். அப்ப இது என்னது 2/2 2/2. அப்ப Wheatstone bridge condition is satisfied. அப்ப நடுவுல இருக்கிற 2 நம்மளுக்கு யூஸ் இல்ல. You can remove it. Once you remove it, these two are in series. These two are in series. அப்ப இது என்ன ஆயிடும்? When they are in series for capacitors, then they are identical in nature. So you'll have to use 1 by C1 plus 1 by C2, or that is 1 by C plus 1 by C. Then inga enna aidu, this 2 by 2 it will come. So it will become 1 microfarad over here. Then similar way you do the series combination formula for the bottom part. Again you will get 1 microfarad. Then these two 1 microfarads are in parallel. In parallel you just have to add the, to find the effective capacitance you just have to add it. So 1 plus 1, it will be your 2 microfarad will be your right answer. The 2 microfarad will be the right answers. You let us move on to the next question. So again a good question to test your understanding. So if you will have to know about the field lines idea here. If generally ஒரு மேக்னெட் அப்படின்னு இருந்தா ஃபீல்ட் லைன்ஸ் எப்படி இருக்கும்? நார்த் டு சவுத் அவுட் சைடு, ரைட்? நார்த் டு சவுத் அவுட் சைடு இப்படி இருக்கும். இப்ப நான் என்ன பண்றேன், இந்த மேக்னெட்டை ரைட்ல நவுத்துறேன். அப்ப ரைட்ல நவுத்துறேன்னா இந்த காயில பொறுத்த மட்டும் இப்படி இருக்கிற மேக்னெட்டிக் ஃபீல்ட் லைன்ஸ் குறையுது. அப்ப அது குறைய விடாது, அது அப்போஸ் பண்ணும். அப்ப இப்படி இருக்கிற மேக்னெட்டிக் ஃபீல்ட் லைன்ஸ் குறையுதுன்னா இன்டியூஸ்டு ஃபீல்டு எப்படி இருக்கணும்? இப்படிதான் இருக்கணும். அப்ப இப்படி இருக்கணும்னா கரண்ட் மேல இருந்து கீழே வரணும்னு அர்த்தம், ரைட்? சோ இது சாரி ஐம் சாரி just a mistake, I'm sorry. So it's north to south, so magnetic field lines are like this. I'm sorry. So north to south the magnetic field lines are like this. So in the field lines இந்த காயிலுக்கு குறையும். நான் இந்த மேக்னெட்டை நவுத்த இந்த லெஃப்ட் டு ரைட் டு லெஃப்ட் ஃபீல்ட் லைன்ஸ் குறையும். அப்படி குறைஞ்சதுன்னா அதை இன்டியூஸ் பண்றதுக்கு கரண்ட் மேல இருந்து கீழே இறங்கும். ஸோ அப்ப ஏல இருந்து பிக்கு வர மாதிரி இருக்கும். ஸோ ஏபி வில் பி த டைரக்ஷன் ஆஃப் கரண்ட் இன் சொலனாய்டு 1. ஏன்னா இந்த ஃபீல்ட் லைன்ஸ் கம்மியாகுது. இப்ப இதுக்கு வருவோம். இதுக்கு வர்றப்ப இந்த இப்ப இதுல இப்படி உள்ள வர ஃபீல்ட் லைன்ஸ் இன்க்ரீஸ் ஆகுது, கரெக்டா? உள்ள வர ஃபீல்ட், இதை நவுத்த உள்ள வர ஃபீல்ட் லைன்ஸ் இன்க்ரீஸ் ஆகுது. இப்போ உள்ள வர ஃபீல்ட் லைன்ஸ் இன்க்ரீஸ் ஆச்சுன்னா அதை அப்போஸ் பண்ணும். அதை அப்போஸ் பண்ணணும்னா இப்படி இருக்கணும். அப்ப இப்படி இருக்கணும்னா எனக்கு கரண்ட் மேல இருந்து கீழே வரணும். அப்ப D to C. அப்ப solenoid 1ல A to B, solenoid 2ல D to C is the right answer. Again a good tricky question, so you have to be a bit careful in the direction of the magnetic fields, right? So let us move on to the next question. Again a theory question. Consider the following statements A and B, identify the correct answer. They have given you 4 quadrants for IV characteristics. The IV characteristics of lies in the 4th quadrant for an solar cell. That would be the question there. So அப்ப சோலார் செல்லோட ஐவி கேரக்டரிஸ்டிக்ஸ் உங்களுக்கு இப்படி இருக்கும். கட் ஆஃப் கரண்ட், ஆல் தட் இஃப் யூ டு சோலார் கேரக்டர் இப்படி இருக்கும். அப்ப இப்படி இருந்ததுன்னா அது எந்த குவாட்ரண்ட்ல இருக்கு? 4th quadrantல இருக்கு. அப்ப ஸ்டேட்மெண்ட் A is right. If statement A is right, then in reverse biased PN junction diode, current measured in microampere is due to majority carriers. No, when it is reverse biased, it is due to minority carriers. So அப்ப A is right, but B is wrong. So again option 3 would be the right answer. Option 3 would be the right answer. Again a question to test your theoretical idea. So let us move on to the next question now. 15th question, again very tricky question. So they have given this as 90 degree. A light ray enters through a right angled prism at point P with an angle of incidence 30 degree as shown. If it travels through the prism parallel to the base BE and emerges along face AC, right? So it's like this, your final refracted rays like this, then the refractive index. அப்ப final refracted ray இப்படி இருக்குன்னா என்ன நடக்குதுன்னு அர்த்தம்? TIR, that is total internal reflection நடக்குதுன்னு அர்த்தம். அப்ப total internal reflectionனா இப்ப இந்த ஆங்கிள் தெரியணும். இந்த ஆங்கிள் ஏதோ நம்மளுக்கு தெரிஞ்சா that should be your critical angle, கரெக்டா? That should be your critical angle. அப்ப இது 30 ன்னு வச்சுப்போம், இது some தீட்டான்னு வச்சுப்போம், ரைட்? சாரி, இதை தீட்டான்னு வச்சுப்போம், இதை தீட்டான்னு வச்சுப்போம். இந்த ரெண்டு ஆங்கிளும் 90. அப்ப இது 90 minus தீட்டா. இது 90 minus தீட்டானா இது தீட்டா, இது தீட்டானா இது 90 minus தீட்டா, கரெக்டா? சோ அப்ப இந்த 90 minus தீட்டா என்னவா இருக்கணும்? உங்களுக்கு கிரிட்டிக்கல் ஆங்கிளா இருக்கணும். இந்த 90 minus theta உங்களுக்கு கிரிட்டிக்கல் ஆங்கிளா இருக்கணும். இப்போ இந்த சர்ஃபேஸுக்கு ஏபிக்கு நான் நார்மல் ஸ்னெல்ஸ் லா யூஸ் பண்றேன். ஸ்னெல்ஸ் லா யூஸ் பண்ணா எனக்கு என்ன வரும்? sin 30, அதோட ஆங்கிள் ஆஃப் இன்சிடென்ஸ் என்ன? 30 by sin r, அதுதான் இதுக்கு theta, right? So because this is the angle of refraction for the first surface is equal to mu by 1 அப்படி சொல்லலாமா? ஸோ அப்ப இதுல இருந்து நான் sin thetaவை 1/2 mu ன்னு வரும். 1/2 mu will be your sin theta over here, right? இப்போ இங்க வாங்க, இங்க வர்றப்ப sin, if 90 minus theta is IC, then sin 90 minus theta should be equal to sin IC, correct? What is sin IC formula? 1/mu. What's sin 90 minus theta? Cos theta, right? நம்மளுக்கு sin theta தெரியும். How do you find cos theta then? So அப்ப இது, this is nothing but your opposite. இதை நீங்க hypotenuse எடுத்துக்கலாம். இது adjacent by hypotenuse. So when you use Pythagoras theorem and do, what you get is, you get 4 mu squared minus 1 by 2 mu will be your cos theta. That should be equal to 1/mu. Now everything is in mu, square on both sides, and the mu cancel out, square on both sides you do, then you simplify, what you get is your option 4 would be the right answer. Root 5/2 would be the right answer. A very good question, one of the tougher questions in this lot. A good question. If you're getting it right, pat on your back. Very good question though. Next question, given below are two statements, one is labeled as assertion and other labeled as reason. The potential, they are saying potential at 2-meter distance is 9 into 10 to the power 3, and they say potential on axial line of dipole is 2p by 4 pi epsilon r square. When you do axial line la potential உங்களுக்கு என்ன வரும்? p by 4 pi epsilon naught r squared will be your potential formula on the axial line. அப்ப இந்த ஸ்டேட் டைரக்டா தப்பு. உங்களுக்கு அந்த பொட்டன்ஷியல் ஃபார்முலா தெரிஞ்சிருந்ததுன்னா டைரக்டா இது தப்பு. நீங்க இந்த இது கொடுத்துட்டாங்க, டைபோல் மொமெண்ட் கொடுத்தாச்சு, ஆர் வேல்யூ கொடுத்தாச்சு, 1/4 pi epsilon naught கொடுத்தாச்சு, சப்ஸ்டிட்யூட் பண்ணி சிம்பிளை பண்ணீங்கன்னா உங்களுக்கு அழகா 9 into 10 to the power 3 ன்னு வரும். அப்ப என்ன அர்த்தம்? அசர்ஷன் கரெக்ட், ரீசன் தப்பு. So assertion is right and reason is wrong. So A is true and R is false is the right answer. So option 1 in this case is the right answer, right? Now let us move on to the next question, 17th question now. The moment of inertia of a thin rod. So the moment you see the rod, then moment of inertia formula tuck 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 recollect பண்ணிருக்கணும். About an axis passing through its midpoint and perpendicular to the rod. So what is the moment of inertia formula for a rod about an axis passing through center and perpendicular to it? It is ml squared by 12, right? இங்க என்னன்னா I என்ன கொடுத்துறாங்க? 2400 gram per centimeter cube, mass 400, ஆன்சர் சென்டிமீட்டர்ல இருக்கு. இங்க நீங்க தேவையில்லாம கன்வெர்ட் பண்ணி மாட்டிக்காதீங்க. எல்லாமே சிஜிஎஸ் யூனிட்ல தான் இருக்கு. உங்களுக்கு ஐ சிஜிஎஸ் யூனிட்ல இருக்கு, லென்த், மாஸ் எல்லாமே சிஜிஎஸ் யூனிட்ல தான் கொடுத்துருக்காங்க. அப்ப நீங்க திரும்ப கன்வெர்ட் பண்ணி ரீகன்வெர்ட் பண்ணணும்னு அவசியம் இல்லை. அப்படியே டைரக்டா தூக்கி I value 2400, then m value 400, L squared by 12 simplify பண்ணி பண்ணுங்க, you will get 8.5 cm. You will get L square as 72 ன்னு வரும். இப்போ 70 L square 72 னா அது 8 க்கும் 9 க்கும் நடுவுல. ஏன்னா L square if it is 72, then L value should be between 8 and 9. ஒரே ஆப்ஷன் தான் இருக்கு, டக் டக்குன்னு மார்க் பண்ணிட்டு போயிட்டே இருக்கலாம். So 8.5 would be the right answer. Is that clear? Reading it. So let us move on to the next question. The terminal voltage of the battery whose EMF is 10 volt and the internal resistance is 1 ohm when connected to an external resistance of 4 ohm is as shown. So this is what is given. So terminal velocity formula பார்த்தோன்னே you'll have to remember there are different forms. The one such form is E minus IR, where R is the internal resistance, E value ன்றது this EMF value. So we need I value first. In the I value வேணும் அப்படின்னா, if you consider as a circuit this 4 and 1 are in series, so it will be E by R plus R. So 10 by 5 it will be 2. When you know I as 2, E is 10 minus 2 into 1, you will get as 8 volt. So option 1 in this case will be right answer. That is 8 volt for this question would be right answer though, right? So let's move on to the next question quickly. 19th question, see a lot of theory questions are coming. So a spectral line, see again, if you're a bit smart in this question, you can solve it easily. You don't have to use Rydberg's formula, all that, sir. 1 by lambda is R into 1 by n square minus 1 by n. All that is there. But if you're a bit smart, they are all from like final status 2. They are all Balmer series, right? So அப்ப எதுக்கு when the difference in the state is lesser, frequency is higher. Sorry, lesser frequency is lesser. இப்போ frequency less இருந்தா வேவ் லென்த் அதிகமா இருக்கணும். அப்ப எதுக்கு டிஃபரன்ஸ் கம்மியோ அதுக்கு இருக்கிறதுலே அதிக வேவ் லென்த். நாலுத்துல ஒன்று தானே இருக்க முடியும். அப்ப இதுக்கு தானே frequency கம்மி. அப்ப இது லைக் டிஃபரன்ஸ் கம்மி, அப்ப வேவ் லென்த் அது. ஓகே. ஸோ அப்ப இது நீங்க ஃபார்முலா படி போட்டீங்கன்னா இட் வில் டேக் லாங் டைம். ஸோ இட்ஸ் a bit smart. இந்த டிஃபரன்ஸ் கம்மியா இருந்தா வேவ் லென்த் அதிகம். அப்ப 3 to 2க்கு இருக்கிறதுலே அதிக வேவ் லென்த். அப்ப ஏக்கு யூ ஷுட் கெட் 3. ஒரே ஆப்ஷன் தான் இருக்கு. ஒரே ஒரு ஆப்ஷன் தான் இருக்கு. நீங்க போட்டு போயிட்டு நீங்க கஷ்டமே பட வேணாம் இந்த கொஸ்டினுக்குலாம். ஸோ அதுக்கு கரஸ்பாண்டிங்கா அடுத்துக்கு என்னது, இட் வில் 486. தென் 5 to 2க்கு இன்னும் கம்மி, இதுக்கு இன்னும் கம்மி. ஸோ தட் வில் பி த ஸோ A3 B4 C2 D1 will be the right answer. Or if you are a bit smart, இந்த கொஸ்டின்லாம் நீங்க சால்வ் பண்ணிலாம் போட வேணாம், ஈஸியா நீங்க போட்டு போயிடலாம். Is that clear? We're getting it. So let us move on to the next. So again another theory questions. Is a lot of theory questions involved in this question paper. If c is the velocity of light in free space, correct statement about photon. So photon theory. So energy of the photon, yes, you can happily accept it. E is equal to h nu. Velocity of photon is c, yes. Then momentum is E by c. E is nothing but h nu. So h nu by c. This is also right. In a photon electron collision, both energy and momentum has to be conserved. Yes, absolutely a correct statement. But here photon possesses a positive charge, is right. It is a chargeless particle though. So this E statement is wrong. All others are right. So A, B, C, and D are the correct right statement in these 5 statements. A good question though. Again, like if you know the photon theory or the basic postulates தெரிஞ்சதுனா சூப்பரா இந்த क्वेश्चन அழகா 30 seconds குள்ள போட்டு போயிட்டே இருக்கலாம். Fine. So next question. So again a question from your nuclear physics here. So X, it undergoes an alpha decay, then it becomes Y. So alpha decay enna agum, mass number will reduce by 4. So about 286. Then atomic number will reduce by 2. Appo inge 80. Then all this E plus abdina, there it's called as beta plus decay. Appo beta plus decay la proton neutron convert agum. So when proton is converted to your neutron, then your mass atomic number will reduce by 1. So 79. Mass number will not change. So 286. Beta minus decay, opposite will happen. So neutron will get converted to proton. So 79 will become 80. There will be no change in mass number. So 286. Again, E minus and beta minus are nothing but the same. They represent beta minus decay. So this is again 286, no change. There will be increase in atomic number by 1. So it will be 286, 81. So this is 286, 81 will be the right answer though. Oh, sorry. So 286, 81 will be the right answer. I'm sorry. So 286, 81 will be the right answer though. Right. So next question. At any instant time t, the displacement of any particle is given by 2t minus 1, and then under the influence of force of 5 Newton. Value of instantaneous power. Instantaneous power. Sir. Instantaneous power. The moment you see instantaneous power, it is F into V. It should be F dot D, but here they didn't mention anything about direction. So F into V. So F5 kudutachu. Displacement kudutachu. Displacement is 2t minus 1. So what about V? V is ds by dt. When you differentiate, you get 2. So force is 5. V is 2. So 5 into 2, which is 10 watt, is going to be the right answer. 10 watt is going to be the right answer. Again, a simple question if you know the basic formula of instantaneous power. Right. So let us move on to the next question. So if this is A, then this will be A bar, because this will be A dot A, the whole bar, and this will be B bar. So here if you have A bar, then this is an OR gate, right? This is an OR gate. So A bar plus B bar, the whole bar will be the final output. A bar plus B bar, the whole bar. So if you use Boolean algebra or De Morgan's theorem, it will be A double bar dot B double bar. A double bar is A, so A dot B. So A dot B is nothing but your AND gate. So option 2 is going to be the right answer. Option 2 is going to be the right answer. Again, logic gate liye simple question na, but compared to the previous question is considered tricky. But logic gates should be a cakewalk for you. Let us move on to the next question of the mass of the planet is 1/10 of the Earth and diameter is half that of the Earth. The acceleration due to gravity on that planet. So on the surface of the planet. So what is the formula of G? at the surface that is relating mass and the radius of the diameter. It will be gm by r squared. So இதுதான். You remember this? அவ்வளவு இந்த question ஒன்னும் அதுக்கு மேல பெரிய கஷ்டமான கொஸ்டின்லாம் கிடையாது. m is becoming 1/10, r is becoming diameter half ஆச்சுனா radius half தான் ஆகும். So அப்ப g dash is nothing but g into m by 10 r squared. So it will be r squared by 4. So if you do so, it will be 2 by 5 into gm by r squared. In the gm by r squared for earth என்னது 9.8. So if you do so, if you 2 by 5 into 9.8, you will get that as 3.92. Again, a basic question from the chapter gravitation. Let us move on to the next question now. So 25th question, again a theory question. Given below are two statements. Statement 1, atoms are electrically neutral as they contain equal number of positive and negative charges. Absolutely right. Right. Then statement 2, when it comes, atoms of each element are stable and emit their characteristic spectrum. The statement may be a bit confusing, but there are very similar lines that are present in your physics NCERT atoms chapter. So statement 1 and statement 2 both are correct. So both statement 1 and statement 2 are correct. And there are they have framed it in a bit tricky way, but there are very similar statements in your physics NCERT. So this statement 2 is also right over there. Right. Let us move to the next question. Again, a question from rolling motion. A wheel of a bullock cart is rolling on a level road as shown in the figure. If its linear speed is v in the direction shown, which of the following options is correct? So now if this p is the topmost point, so what is the velocity or the speed of the topmost point will be your linear speed plus r omega. For bottommost point, it is linear speed minus r omega. They didn't mention about pure rolling also, but it is, you can easily see here, because it is combination of translation plus rotation of v plus something, v minus something. Final conclusion easy on the point p moves faster than q. Point p moves because velocity speed of point p is v plus r omega, q v minus r omega. So point p moves faster than q would be the right answer in this case. So let us go to the next question. 27th question, a particle moving with uniform speed in circular path. Again, a very trapping question. Most of you will fall for the trap. So please be careful. In a circular motion, if speed is remaining constant, not necessarily velocity should remain constant. Why? Because direction will keep on changing. At this point, direction will be this way. Direction will be this way at this point. So as you can see, even though the magnitude of velocity remains the same, but the direction will keep on changing. Direction மாறிட்டே இருக்கு. Direction மாறினா constant velocity சொல்லக்கூடாது. Direction மாறினாலோ magnitude மாறினாலோ அது constant நீங்க சொல்லக்கூடாது. So அதே மாதிரி பாத்தீங்கன்னா varying velocity. அதே மாதிரி இது velocity direction. Acceleration பார்க்கறப்ப they are all pointed towards the then அப்ப at every instant the direction of acceleration changes. Magnitude is v squared by r சொல்லலாம் constant. Direction பார்க்கறப்ப மாறிட்டே இருக்கு. As you see from the ground frame, it is keep on changing. So since direction is changing for velocity and acceleration, it has varying velocity and varying acceleration. Equal speed, uniform speed னு பார்த்தோம்னா constant velocity போட்றாதீங்க, direction மாறுதீங்க. Very good question again. So let's move on to the next question quickly. So a thin flat circular disc of radius 4.5 centimeter is placed gently over the surface of the water. If the surface tension of water is 0.07, then the excess force required. So a new model of question introduced. You will not see much of this kind of questions in the previous year NEET. In the NEET la pudu model introduced ஆயிருக்கு. So your surface tension is force per unit length. So force, that extra force is due to surface tension. So surface tension into length. ஒரு டிஸ்க் அப்படின்னு பாக்குறப்ப இந்த ஃபோர்ஸ் அதோட சர்க்கம்ஃபரன்ஸ்ல ஆக்ட் பண்ணும். அப்ப லென்த் இடத்துல என்ன எடுக்கணும்? 2πr எடுக்கணும். அந்த எக்ஸ்ட்ரா ஃபோர்ஸ்ன்றது சர்ஃபேஸ் டென்ஷன் இன்டூ 2 இன்டூ பை இன்டூ ரேடியஸ். சர்ஃபேஸ் டென்ஷன் வேல்யூ கொடுத்தாச்சு. பை வேல்யூ 22/7 எடுங்க, 7 கேன்சல் ஆயிடும். r வேல்யூ கொடுத்தாச்சு. when you substitute and simplify, you will get the answer as, I think it is 19.8. So you'll get the answer as 19.8 Newton. Those option 3 here will be the right answer. So a new model of question introduced here. We can't see much of this question in the previous year questions. Right, fine. Let us move on to the next question. 29. Again, a question from your magnetism part. Again, a bit complicated. You have to know the idea behind it. When you have a dipole oscillating in a magnetic field, you will have to know this formula. Otherwise, you can't go about it. It is 2 pi root of I by mb. It is 2 pi root of I by m into b. So time period, how can you calculate? 20 oscillations in 5 seconds. Now one oscillation will take 1 by 4 seconds. So time period is 1 by 4 seconds. I value கொடுத்தாச்சு, m value கொடுத்தாச்சு. M value in terms of x இருக்கு, b value கொடுத்தாச்சு. Just square it. Then your root will go அப்பயே நீங்க என்ன பாதி அப்புறம் பயம் போயிடும். இந்த ரூட் இருந்தாலும் நம்மலாம் பயப்படுவோம். ஸ்கொயர் பண்ணிருங்க. அப்ப அது என்ன ஆயிடும்? 4 pi squared into I by mb ஆயிடும். இப்ப எல்லா வேல்யூ இருக்கு. இனிமே you just do the basic simplification. You'll get answer as 1280 pi squared. 1280 pi squared will be the correct answer over here. Again, a bit tricky question. It's not that easy question, though you'll have to know this logic, otherwise it's a tough question. So let us move on to the next question now. 30, a simple question again. Two bodies A and B of same mass undergo completely inelastic. So completely inelastic na என்னது? கொலிஷனுக்கு அப்புறமா ரெண்டும் ஒரே வெலாசிட்டில போகுதுன்னு அர்த்தம். கொலிஷனுக்கு அப்புறமா ரெண்டும் ஒரே வெலாசிட்டில போகுதுன்னு அர்த்தம். That is the clue. Body A moves with velocity v1 while body B is at rest before collision. அப்ப conservation of momentum படி ரெண்டுத்துக்கும் சேம் மாஸ், அப்ப m ன்னு எடுத்துக்கலாம். m into v1 plus m into 0, that is before collision momentum. That should be equal to after collision என்னது mass. Like both the mass will have same velocity, அப்ப 2m into v2 அப்படின்னு சொல்லிருக்காங்க. Right? Now if you see v1 by v2 போட்டீங்கன்னா அழகா என்ன வரும்? 2:1 அப்படின்னு வரும். v1 by v2 you do, then you will get 2:1. So option 4 is going to be the right answer in this case. Again, a pretty simple question if you are clear with the collisions part. So let us move on to the next question. So 31, again a very simple question. If you are good in your SHM part, what is the general equation of an SHM? X is equal to a sin omega t plus phi. So அப்படியே இந்த ஜெனரல் ஈக்குவேஷனோட கம்பேர் பண்ணுங்க. ஏ இடத்துல யார் இருக்கா? 5. அவ்வளவுதான். ஆம்ப்ளிடியூட் இஸ் 5. ரைட். So they have given in meters, so அது 5 meters தான். அப்ப இந்த ரெண்டு ஆப்ஷனை எலிமினேட் பண்ணிடலாம். Then what you are supposed to find? time period. time periodக்கு ஒமேகா தெரியும். அப்ப இத கம்பேர் பண்ணீங்கன்னா ஒமேகா இடத்துல யார் இருக்கா? ஒமேகா இடத்துல pi. So omega is nothing but pi. Right? Omega is nothing but pi over there. அப்ப ஒமேகா எப்படி எழுதலாம்? 2 pi by time period. So 2 pi by time period is pi. Pi goes off. The time period is 2 seconds. So 5 meter, 2 seconds will be the right answer. So option 4 in this case will be the right answer. Is that clear? So let us move on to the next question. The quantities which have same dimensions as those of solid angle. Solid angle doesn't have dimension. That is the catch over here. Appa you'll have to check options which doesn't have dimensions. Appa strain dimension kaniyadu. Solid angle, angular dimension again. So strain and angle will not have any dimension. So the answer would be option 3 in this case. Is that clear? So let us move on to the next question. Again, if you are good in electrostatics, if you understand that simple idea that it is a thin spherical shell, apply the hollow sphere. In a hollow sphere, your potential is constant through inside. Outside it will vary. So whatever is the potential on the surface will be the potential anywhere you take it inside. But they are asking potential difference. They are not asking potential. So whatever is the potential at P will be, so Vp will be equal to Vc, because in an hollow sphere, potential inside will be same, and that will be equal to potential on the surface. அப்ப இதுல இருந்து Vp minus Vc என்ன ஆயிடும்? 0. So the potential difference will be 0. So option 2 will be the right answer. Again a simple question. If you are on the basic results எல்லாம் நம்ம தெளிவா எலக்ட்ரோஸ்டாட்டிக்ஸ் இருக்கிற ரிசல்ட்ஸ் எல்லாம் நமக்கு தெளிவா இருந்தாலே இந்த क्वेश्चन கஷ்டப்படாம அட்டெண்ட் பண்ணிட்டு போயிடலாம்னு அர்த்தம். Clear about it? So let us move on to the next question, 34. So a bob is whirled in an horizontal plane by means of a string with an initial speed of omega. So when omega is involved, please be careful, your tension acts as centripetal. That is clue number 1. When omega is involved, please ever don't take mv square by r. When omega is involved, then it is mr omega squared. இப்போ என்ன சொல்றாங்க? Omega 2 omega is becoming 2 omega. அப்ப tension it is square, right? Mass மாறல, radius மாறலன்னு சொல்லியாச்சு. அப்ப omega 2 omega மாறறப்ப புது டென்ஷன் என்ன ஆயிடும்? mr into 4 omega square ஆயிடும். So t dash is nothing but 4 times the new tension will be 4 times the old tension. 4t will be the right answer in this case. Again a very simple question if you are good in your circular motion. Basic idea of circular motion is enough, not complicated concepts are not needed here. Okay, let's move on to the next question now. 35 questions. So we are coming to the end of section A here. We'll quickly do it. So wire of length L and resistance 100 ohm is divided into 10 equal parts. So அப்ப 10 equal parts na each part oda resistance என்னவா இருக்கும் 10 ohm because it is resistance is proportional to length. மொத்தமா 100 அதை நான் 10ஆ பிரிக்கிறேன். அப்ப ஒவ்வொரு பார்ட்டுக்கு எனக்கு என்ன இருக்கும் 10 ஓம் இருக்கும். அப்புறம் என்ன சொல்றாங்க first 5 parts are connected in series. அப்ப 5 பார்ட்டை நான் சீரிஸ்ல கனெக்ட் பண்றேன். சீரிஸ்னா என்ன பண்ணலாம் effective will become or you should add it. அப்ப இதோட மொத்த effective என்ன ஆயிடும் 5 ohm அப்படின்னு ஆயிடும். அதுக்கு அடுத்து என்ன சொல்றாங்க 5 நான் parallel ல கனெக்ட் பண்றேன். they are saying another 5 pieces are connected in parallel, right? So அப்ப parallel ல identical க்கு it is r by n. If n identical resistance are connected in parallel then r by n இது எல்லாமே 10. அப்ப 10 10 10 10 10 இருந்தா இதோட effective என்ன ஆயிடும் 2 ன்னு ஆயிடும் 10 by 5. So it will become 2. So then so this is 50. When you add it will become 50. So இது ஒரு 50 எல்லாத்தையும் இது 10 10 எல்லாம் ஆட் பண்ணீங்கன்னா it will come as 50. Then இதோட parallel if you take it will be 2. So then they are again connected in series. So it will be 50 plus 2 which is 52 ohm. Again a good question to give us an idea about this series parallel combination. So first 5 series அப்ப அதுக்கு 50, அடுத்த 5 parallel அப்ப அதுக்கு effective 2. இது ரெண்டும் சீரிஸ்ல இருக்கு. அப்ப 50 plus 2 52. So 52 will be the right answer in this case. So that's the end of your section A. Let us quickly move on to section B. There is no much difference in the standard of questions asked in section A and section B. So even there are few easy questions in section B as well. So இதுல TV graph கொடுத்திருக்காங்க. So we know PV is equal to nRT. So I can write T is equal to P by nR into V. So if you see the slope is directly proportional to pressure. You can see that slope is directly proportional to pressure. So whichever is having a higher slope will have a higher pressure. The மூணுக்கு யாருக்கு slope அதிகம் P1 க்கு தான் slope அதிகம். அப்ப P1 ஓட value தான் அதிகம். So P1 greater than P2 greater than P3. So higher the slope, higher will be the pressure in TV graph. So option 2 here in this case is the right answer. So again a simple question. ஆனா இந்த மொத்த கொஸ்டின் பேப்பருமே எப்படி இருக்கு சின்ன சின்ன பேசிக் திங்ஸ் எல்லாம் நம்ம கரெக்டா ரீகலெக்ட் பண்ணோம்னா சூப்பர் மார்க் இதுல ஸ்கோர் பண்ணலாம். ரைட் ஃபைன். So moving on to the next question now. A parallel plate capacitor is charged by connecting to a battery resistor. The if I is the current in the circuit then gap between the plates. So அப்ப is charged அப்படின்னா அப்ப அந்த மூவ்மெண்ட் ஆஃப் சார்ஜஸ் இருக்குன்னு அர்த்தம். அப்ப கரண்ட் இருக்குன்னு அர்த்தம். இப்போ ஒரு வெளியில கரண்ட் இருக்கிறப்ப ஒரு சார்ஜிங் கெபாசிட்டர்ல வெளியில கரண்ட் இருக்கிறப்ப உள்ள there will be something called as displacement current. That is what is Maxwell says. So அப்ப அந்த displacement current நேச்சர் எப்படி இருக்கும்னா it will be having the same magnitude as that of your conduction current and it will be in the same direction as that of your conduction. உள்ள ஒரு சின்ன கரண்ட் இருக்கிற மாதிரி அது ஆக்சுவலா இல்ல பட் இருக்கிற மாதிரி நம்ம அசியூம் பண்ணிக்கலாம். So it will have a property similar to that of your conduction current. So option 4 in this case is the right answer. So moving on to the next question. The property which is not for an electromagnetic wave traveling in free space. They travel in free space nu kudutaachu. So they travel with the speed equal to 1 by square root of mu naught epsilon. Yes, it's absolutely right. But inga enna kekranga not a property kekranga. So they originate from charges moving with uniform speed. Here is where the twist is. Your electromagnetic waves are generated when the charges are accelerated. So அவங்க uniform speed சொல்லிட்டாங்க, which means it is not accelerating. So this will not generate EM wave at all. They are transverse in nature. Yes, they are right. Energy density is equally distributed in electric field and magnetic field. Yes, it is absolutely right. So in the second statement is not an property of your electromagnetic wave. Right. So moving on to the next question now. So again a question bit tricky in nature. So here they have given which can achieve bridge balance here. So இது எல்லாமே 10 15. அப்ப இங்கேயும் எல்லாமே 10. அப்ப ஐடியா இஸ் இங்க 15 வரணும். இங்க 15 வரணும் அப்படின்னா இப்ப இதுல பாத்தீங்கன்னா அவங்க இது மாத்திட்டாங்க. They have changed the polarity. So this will become reverse biased. அப்ப இதுல current flow இந்த இதுல இருக்காது. Same happens over here. This is reverse biased. So current flow இருக்காது. உங்களுக்கு இந்த 5 15 மாறணும்னா it's a diode. They didn't say it's ideal also. So this diode can add up the resistance. So if diode is having a resistance, forward resistance of 10 ohm, then this can possibly act as an bridge balance. They are not asking which will, which can is only is asked. So option 3 in this case would be the right answer. ஏன்னா இங்க diode நம்மளுக்கு அந்த எக்ஸ்ட்ரா ரெசிஸ்டன்ஸ் ப்ரொவைட் பண்ற சாத்தியக்கூறு உண்டு. They can provide that extra resistance. So this can act, this may not, but this can act as an bridge balance, that is Wheatstone bridge balance. That is clear? A good question. If you're confusion ஆனா section B இதெல்லாம் நீங்க பார்த்தோம்னா கன்ஃபியூஷனா இருந்தா என்ன பண்ணிரணும், you just leave it because it's a bit confusing. Fine. So coming to the next question, 40. If the plates of the parallel plate capacitor connected to a battery, this is the clue over here, connected to a battery. So as long as the capacitors are connected to the battery, என்ன அர்த்தம், அதோட potential difference மாறாது, அதோட potential difference மாறாது. So your V will remain constant. That is clue number 1. அடுத்து என்ன சொல்றாங்க, they are moved closer. What is C formula for parallel plate capacitor? This is epsilon naught A by D. When D decreases, your C will increase, right? So capacitance increases, right? Then we have Q is equal to CV. V doesn't change because it is connected to battery. C increase aachana Q increase agum, charge stored in it increases, right? Then energy formula enadhu for a capacitor, half C V squared. V constant na C increase ana EU increase aganum, but inga enna sollirukanga, decreases nu sollirukanga. Statement B தப்பு. அடுத்து ratio of charge to its potential, அதாவது Q by V ன்னு கேக்குறாங்க. V same, Q மாறும், it will not remain the same, இது தப்பு. Q by V ல V மாறாது, ஆனா Q மாறுது, Q increase ஆகுது, increase ஆகுதுன்னா அந்த Q by V சேமா இருக்காது. அடுத்து என்ன சொல்றாங்க, product of charge and voltage increases, Q into V மாறாது, Q ஏறுது increase ஆகுதுன்னா ப்ராடக்ட்டும் increase ஆகும். E statement is right. So A, C, E are the correct statement over here. Again, a good question to give us an understanding of your capacitor, basic understanding. This is clue. இத புடிச்சு இந்த C value புடிச்சிட்டீங்க அப்படின்னா இதே கடகடகடன்னு மத்தெல்லாம் cross check பண்ணிட்டு போயிட்டே இருக்கலாம்னு அர்த்தம், right? So let us move on to the next question now. 41. The force is defined by F is equal to alpha T square plus beta T acts on the particle at a given time T. The factor which is dimensionless if alpha and beta are constant. So the idea is each term, if you write an equation, each term should have the same dimension. Alpha T squared should have the dimensions of force. Then beta T should have the dimensions of force again. Right? So அப்படி பாக்குறப்ப force dimension என்ன, mlt power minus 2. So அப்படி போடுறப்ப alpha ஓட dimension என்ன வரும், or mlt power minus 4 னு வரும். Beta order dimension you'll get mlt power minus 3. So when you multiply T with alpha, then it will become the dimensions of beta. So alpha T by beta will be dimensionless. So they are asking what is dimensionless. So alpha is kandupidichi, beta is kandupidichi, or you can simply divide these two, then you will get the answer. The other way of doing it is divide these two, then right hand side will become dimensionless. Alpha T by beta abdin varum. So in that way also you can do. So option 4 is the right answer in this case. Moving on to the next question. 42. A metallic bar of Young's modulus 0.5 10 to the power 11 and coefficient of linear expansion 10 to the power minus 5. Length 1 meter, area is given, it is heated from 0 to 100, temperature difference is given. They are relating, asking us compressive force. If you are an Allen student, it is a direct question from your module. So here in this case, what we know, your y is F into L by A into del L, right? F into L by A into del L. From our thermal physics or thermal expansion, we know del L by L is alpha del T. So in the del L by L, when you do F by A into alpha del T, you will get. So F is nothing but F in y into A into del T into alpha. Y value is given, A value is given, del T value is given, alpha value is given. So just substitute and simplify, you will get 50 into 10 to the power 3 as the correct answer. Again, a good question that connects your mechanical properties and your thermal physics. Konja and the application factor is that it's like a moderate question. So 15 to the power 3 will be the right answer in this case, right? So 43, a small telescope has an objective of focal length 140 and an eyepiece of focal length, so this will not be, and an eyepiece of focal length 5 centimeter. The magnifying power of a telescope for viewing a distant object. So direct question, magnifying power is F0 by Fe. So it will be 140 by 5, which is 28. So F naught by Fp, so 145y, which is 28 in this case. So option 4 is the right answer in this case. So now moving on to the next question. An iron bar of length L has a magnetic moment M. It is bent in the middle of its length such that the two arms make an angle 60 degree with each other. Magnetic moment of this new magnet. Again, a question from magnetism. See what initially you have one magnet where they say this M, we know its pole strength. Let me call that pole strength as Q now. So Q into L. Whatever M they have given is Q into L. Now what they say, they are bending at 60 degree. So this will become L by 2, this will become L by 2, right? Because they are bending at the midpoint. So this is L by 2, this is L by 2. It is 60. If they both are same, then this length will again be L by 2. So when you calculate the magnetic moment, it is pole strength. By bending, pole strength will not change. It is only effective distance between the two ends. So effective distance between two ends is again L by 2. What is QL? QL is M. So it is M by 2. So option 4 is going to be the correct answer. So here what you have to remember is magnetic moment is pole strength into the effective length between the two ends. So if you can find this effective length, then it will be L by 2. So it will be QL by 2. QL is again M. So M by 2 will be the right answer though. So now moving on to the next question. A 10 microfarad capacitor is connected to 210-volt 50 hertz source as shown. The peak current means maximum current. So here I naught is E naught by XC in this case. XC is 1 by omega C. But I naught is omega C into E naught. Here whatever 210 is not the peak voltage. It is the RMS voltage. அப்ப இங்க தான் இந்த क्वेश्चनக்கான கேட்ச் எங்க இருக்கு? இந்த இடத்துல இருக்கு. This is ERMS. அப்ப E naught வேணும்னா என்ன பண்ணணும்? ERMS into root 2. So omega is 2 pi into frequency. Frequency is 50 hertz. C value கொடுத்தாச்சு. ERMS 210 root 2 value தெரியும். When you substitute, what you get is 0.93 ampere. Again, a good question. Just a simple conversion from ERMS to peak value should be that. Then you'll get the answer perfectly. Now moving on to the next question. Two heaters A and B have power rating 1 kilowatt and 2 kilowatt respectively. Those two are first connected in series and then parallel source. Again, bit of a complicated question. See here the source is fixed. So when source is fixed, your V is fixed. When V is fixed, your resistance is inversely proportional to power. So you can say your R1 by R2 will be like 2:1 or 1:2, whatever you want you call. Now what they say, they first are connected in series. So power 1 will be V squared by, when they are in series, R1 plus R2. Then power 2 will be V squared by R effective. Here 1 by R effective is 1 by R1 plus 1 by R2. So V squared into 1 by R1 plus 1 by R2. What they are asking here? இதோட ratio கேக்குறாங்க. P1 by P2 கேக்குறாங்க. அப்ப P1 by V2 கேட்டா same source, அப்ப V will cancel. When you simplify, what you get is something like this: R1 R2 by R1 plus R2 the whole square. Again, when ratio is asked, in the value update எடுத்து போடலாம். For mathematical simplicity, logically is not supposed to. When you do that, then what you get is 2:, and when you do that, what you get is 2:9. So option 4 is going to be the correct answer in this case. Let us move on to the next question. We are almost to the end. I know I am going a bit fast. So we are running short of time. So 47th question. VT graph of motion of a body is shown. Our VT graph கொடுத்தாச்சு. Initial velocity இல்ல. அப்ப initial acceleration இருக்காது. அப்புறம் velocity, it's a straight line with a constant slope. Slope of velocity time acceleration கொடுக்கும். இங்க slope constant, அப்ப acceleration constant. அப்ப கொஞ்ச டைம்க்கு அப்புறமா இந்த டைம் some t1 to t2, your acceleration should be constant. அப்ப இங்க constant இருக்கு. Then from t2 to, let it be t3. From t2 to t3, your velocity is constant. When velocity is constant, what about acceleration? 0. அப்ப t2 to t3 உங்களுக்கு 0. அதுக்கு அடுத்து negative slope, so negative acceleration. So you will have, again, slope is constant, so acceleration will also be constant. Their option 1 perfectly represents the AT graph of this given VT graph. Again, a good question to test your graphical analysis or graphical knowledge. So let us move on to the next question. 48. If mass of the bob in a simple pendulum is increased to thrice its original mass and length is made half of its original, then new time period is in here. The catch is Your time period, what is the time period of simple film? 2 pi root of L by G. It is independent of mass. அப்ப அந்த 3 times increase ஆகுது கொடுத்திருக்கிற டேட்டா இங்க தேவையில்லை. இப்போ new time period என்னது? New time period will be 2 pi root of length is becoming half of it. So L by 2G. So I can write it as root 2 pi root of L by G. இந்த T dash என்ன கொடுத்திருக்காங்க? X by 2 times the original value. Original value is 2 pi root of L by G. So 2 pi root of L by G, 2 pi root of L by G goes off. நம்மளுக்கு என்ன கேக்குறாங்க? Root 2 will be in the denominator. So what they are asking x value. So x will be 2 by root 2, which is root 2. So option 4 is going to be the correct answer in this case. So the catch here is, if you are concentrating more on the mass part, mass கொடுத்திருக்காங்களே 3 times அதை எங்க யூஸ் பண்றது அப்படின்னு பாத்தீங்க அப்படின்னா it will not work here because time period is independent of the mass of the bob. So only length matters here. Length half ஆகுது, use the correct formula, then do some basic maths, you will arrive at the answer within 30-40 seconds. So this question should not take much time. So moving on to the next question. The minimum energy required to launch a satellite of mass m from the surface of the earth of mass m and radius r in a circular orbit at an altitude 2r. Again, a tricky question from your gravitation. First, it is at the surface. அப்ப surfaceல இருக்கிறப்ப it is only potential. The total energy will be only potential energy in this case. So initial energy will be minus gmm by r. Then it is going to circular orbit. When it is going to a circular orbit, then the total energy is minus gmm by 2 into its orbital radius. So E final will be minus gmm by 6r because 2 into 3r, 2r from the surface, and from the center it will become 3r. So there are all the catchy points here. A good tricky question here. So you have to calculate the circular orbit radius. And the circular orbit radius கண்டுபிடிக்கணும். 2r ன்னு எடுத்துட்டீங்கன்னா போச்சு. 2r is only from the surface, but from the center it is 3r. Then you do E final minus E initial, then you will get option 3 as here, and that is what the energy required. So your E final minus E initial, when you do so, you will get 5 gmm by 6r as the right answer. Right, again a good question from gravitation. The final question of this paper, question number 50. A sheet is placed on a horizontal surface in front of a strong magnetic pole. Force is needed. எதுக்கெல்லாம் ஃபோர்ஸ் வேணும்னு கேக்குறாங்க. To hold the magnet if it is magnetic. Yes, heat magnetic இருந்தா அது it will either attract or repel, doesn't matter. அது ஒரு ஃபோர்ஸ் வேணும். நான் மேக்னெட்டிக்கா இருந்தா பிரச்சனை இல்லை. ஸோ இதுக்கு ஃபோர்ஸ் தேவையில்லை. Move the sheet away from the pole. So if you are moving the sheet, it's like charged conductor, right? So conductor has free electrons. So charged particle moving in a magnetic field in கணக்காயிடும். So when a charged particle is moving in a magnetic field, what happens? It will start experiencing magnetic Lorentz force. அப்ப அங்க Lorentz force is getting involved. So அப்ப you need some force to do this. If it is non-conducting, doesn't matter. It doesn't have your free electrons. So A and C you need force. So option 4 is going to be the correct answer. Right? So with this we have come to the end of this paper discussion. I hope it would have been very useful for you, starting from botany, then zoology, then chemistry and physics. So I hope and wish you all the best. I hope you get most of the questions right and sit in your favorite college. So I wish everyone who is in the class or in this session all the best, and thank you so much.